everyone. We start with the third session of the day. I'm Camilla from the Polytechnic University of Marche, and I'm again the moderator of the session. We have as a moderator Caterina from the University of Marche. From País Vasco, Ana Marie from Ghent University, and Christophe from University of Cottbus. We have Amanda with the thesis of reproductive biology of two deep sea chrysogorgiid corals across the ocean basins. You have 15 minutes. When you have 2 minutes left, I will stand up. The floor is yours.
Hi, I'm Amanda, and this is my thesis on reproductive biology of two deep-sea chrysogorgiids. So first off, to give you some context, the deep sea is characterized by many features, one of the most prominent being seamounts, which can be defined as underwater mountains, at least 1,000 meters raised above the seafloor, and are known biodiversity hotspots. Now, as you can see here, seamounts are very common and globally distributed, though a vast majority of them remain uncharted. Now, seamounts are also known to interact with overlying currents, which results in some complex hydrodynamics, which can increase the upwelling and generate waves that mix the water column nearby it, which also could result in increased surface primary production as well as nutrient availability. So deep sea corals are among the most abundant taxa on seamounts, and they lack zooxanthellates, unlike their shallow water counterparts, so they rely primarily on filter feeding for their nutrients, and the hard substrate and enhanced currents associated with seamounts create ideal environmental conditions for these sessile suspension feeding organisms. Now, these organisms are also of ecological importance as ecosystem engineers, which basically means they create complex three-dimensional habitats that give refuge, spawning grounds, and nursery to diverse invertebrate and fish species. Now, they're also classified as being slow-growing and long-lived, which has in turn led to many deep-sea coral ecosystems being categorized as vulnerable marine ecosystems, which basically just means that they are in need of more protection. So disturbance can result in the loss of these critical habitats, which can take anywhere from decades to centuries to replace. One of these disturbances is bottom contact fisheries. Seamounts are known to host very dense aggregations of demersal fishes, often of commercial interest, and there have been several cases documented of prolonged and intense fishing impacts on seamounts that have resulted in not only the collapse of these fisheries, but also lasting damage to the seamounts. So for example, you can see there's a pretty healthy coral garden on the seamount on the left, and then on the right is extensive seafloor scarring from trawling damage. Another potential impact is deep sea mining, as the minerals of interest are often existing both on and around seamount ecosystems. And in general, for human impacts, there have been several recent policy changes in the United States that have led to the reopening of previously closed fishing grounds on seamounts, as well as several exploration licenses for deep sea mining have already been allocated. So taken together, this underscores the need to collect baseline data now before these ecosystems are irreversibly damaged. So seamounts are pretty patchy habitats, and coral offspring can disperse both on their seamount and between seamounts nearby, as you can see in this little diagram. And understanding the reproductive traits can give us insight into larval dispersal, population connectivity, recovery potential, and resilience to disturbance, all of which are absolutely crucial to understanding if these damaged populations can recover, and if so, how long that may take. So, diving a bit deeper into reproduction, there's supposed to be corals here. Just imagine there's corals here. My PowerPoint got messed up. So deep sea corals can either be hermaphroditic, which means one colony can produce gametes from both sexes, or gonochoric, which means there will be separate male and female colonies. And then they can also produce both asexually and sexually, but asexual strategies are considered rare in the deep sea, so today I will focus on the two primary sexual strategies. So for brooding, you have a male coral here releasing its sperm into the water column, and then a female coral here, and the egg is retained within the female colony, and the sperm fertilizes it on the female colony. And then for broadcast spawning, both gametes are released into the water column instead, where they fertilize there. So this is a diagram of all of the coral reproductive data known to date in the deep sea. Don't worry about reading all of the details. I know it's very overwhelming, but the point is to show you guys that there is a huge knowledge gap with only 4% of species having this trait characterized. Chrysogorgids in particular have had absolutely no reproductive data characterized until today. So my study focuses on 2 chrysogorgids, the first one of which is Metallogorgia melanotrichos, and 9 colonies were sampled along the New England seamount chain from 2004 to 2005. And this classifies them as legacy material, which basically just means that they were collected decades before and not originally intended for my study, but we were still able to extract reproductive data 20 years later. And my second species is Iridogorgia magnispiralis. We only had 1 colony for this one, though it was collected more recently in 2025 from the Marshall Islands. So this thesis aims to determine the reproductive mode and strategy of both of these corals, quantify the gamete characteristics and fecundity, as well as relate this fecundity to local environmental conditions. So the methods used to address these aims are as follows. Our samples were split up and decalcified and then embedded into paraffin wax. And they were then sliced very thinly on this machine called the rotary microtome and then mounted onto microscopic slides, which were then stained using hematoxylin and eosin. And then finally, they were imaged to identify, count, measure, and stage class our gametes. So, female gametes, otherwise known as eggs or oocytes, are broken up into 4 stage classes. The first 2 of which, eugonia and previtellogenic oocytes, are considered immature. And the final 2, vitellogenic, shown here in the circle, and late vitellogenic are considered mature or viable gametes, which basically means if they were released at this point in the stage, they could be fertilized in the wild. And then for male gametes, again, there are 4 stage classes. They're also known as spermatocysts. And shown here are stage 1 and stage 2, the 2 immature classes. And then we have stage 3, our first mature class, and then stage 4. So we also looked at a variety of environmental variables, including productivity, salinity, bathymetry, and current regimes, but I'll circle back to this later when I relate it to fecundity. So the moment you've all been waiting for, what did we find? We found that both corals are gonochoric broadcast spawners, and we found this out by each colony containing either male or female gametes, never both at the same time, which points to gonochorism. And then for broadcast spawning, there were a lot of gametes per polyp, as well as no brooded embryos or larvae present, which is how typical deep sea studies distinguish between brooders and broadcast spawners. And then for size at maturity, all of our colonies were reproductive, so we can give some general estimates with Metallogorgia, our smallest reproductive colony, was 0.6 meters, which means that colonies larger than this will also likely be reproductive. And then for Iridogorgia, colonies around the same size are also likely to be reproductive because it was very large and highly fecund. But obviously more samples are needed for both of these to determine full size at maturity. And yeah, again, corals are supposed to be there. Okay, so let's dive into gamete characteristics. First off, I have some oocyte size distributions here. As you can see, all of the colonies for the females are dominated by small immature stage classes, which is a pretty common pattern in deep sea corals, and this points to a maintenance of a continuous pool of developing gametes. And then as you can see in these 2 specifically, there's some pretty large oocytes as well, with the largest being 670 micrometers. And this points to the development of leucophotrophic larvae, which basically means that they can rely on internal energy reserves rather than feeding in the water column before settling down. And this type of larvae is also consistent with longer larval duration, greater fertilization success, as well as greater dispersal potential. So now for the oocyte stage compositions by colony. As you can see, there are some slight differences among the colonies, but overall, there was no significant difference from the proportion of mature gametes across all colonies. And then same for the male colonies of Metallogorgia, we have some slight variation in stage class composition, but again, no significant difference between the proportion of mature gametes. Now, overall, between both sexes, this is female, this is male, my PowerPoint's messed up, you can see there are multiple stage classes simultaneously, which points to continuous and asynchronous reproduction. Now, keep in mind we only had one time point sample for each sampling period, so we can't fully resolve seasonality in spawning cycles, but with the current data available, the pattern suggests continuous development, but this needs to be investigated further in the future. So moving on to fecundity. We use effective fecundity as our metric here, which can be defined as the number of mature gametes per polyp. So for female fecundity, the 3 in this box are much higher significantly than the 2 being indicated to in arrows. And this variation in fecundity can be due to many factors, anywhere from colony maturity to polyp partitioning, genetics, environmental variables, basically anything. But the one on the far left specifically showed signs of actually transitioning from a juvenile stage to an adult stage based on the polyps centered on the central axis, as these polyps typically disappear when they become fully mature. So that could be a potential reason for why this colony was specifically really low in reproductive output. So for the male colonies, we also had 3 that were significantly higher in fecundity than the one being indicated to. However, this colony did not show signs of transitioning from juvenile to adulthood, so it's more likely that environmental factors, genetics, or polyp partitioning play a role in explaining this lower fecundity here. And to give you a species by species comparison, you can see here that Iridogorgia on the left was much higher in fecundity than the composite of the Metallogorgia samples, which was a pretty surprising thing to see because we expected fecundity to be lower in the oligotrophic Pacific environment versus the highly productive New England seamount chain. However, this is more of like a preliminary interesting finding rather than an actual result because we can't really compare one colony of a different species to a composite of 4 colonies, but it's just fun to look at. Okay, and now for the environmental conditions. We did not find any correlations with salinity, temperature, or any productivity metrics. However, our small sample size and broad-scale productivity data could limit our ability to detect such effects, but regarding my analysis, only the bolded variables showed significant correlations. So for our male Metallogorgia colonies, they actually were more fecund when the seamount was more shallow. And a possible explanation for this is that shallower seamounts often interact more strongly with overlying currents, which again could enhance the nutrient transport and water flow. And then for our female colonies, fecundity was correlated with current speed and direction, which, as we all know now, deep sea corals rely on filter feeding from the water column. So this basically just points to water movement is important for food flow, which in turn addresses reproductive capacity. But currents, the current data that I used was also pretty broad scale, so we don't know what the exact measurements looked like at each location. So in situ measurements in the future would be better to resolve if this affects other colonies, but the pattern is still there in general. So future studies should look at addressing time series data to fully resolve the seasonality and question of if they are continuously and asynchronously reproducing or if there are some more details there. And then also increasing sample size across multiple seamounts and depths would be great. And then, like I said, direct in situ measurements of currents and food flux would help resolve some of the questions and confirm the findings that I have here. As well as genetic connectivity studies would be very useful in determining exactly which populations are feeding other populations through their larval dispersal, and also for highlighting reproductive hotspots in general. So here are some key takeaways I have addressing each of my thesis aims. So for the first one, both species were likely gonochoric, which means, again, separate colonies producing male gametes versus female gametes. And then broadcast spawning, which means they both release their gametes into the water column where they mix and fertilize there. And then our gamete characteristics pointed to continuous reproduction and asynchronous reproduction, with the larger eggs that we found indicative of lecithotrophic larval development, which is the ones that feed within the energy inside them rather than in the water column, like I said. And then finally, only bathymetric and current data were the significant local conditions regarding fecundity. So in the end, this is the very first study that characterized the reproductive traits of these 2 chrysogorgid species, Metallogorgia melanotrichos and Iridogorgia magnispiralis. And our highly fecund colonies are likely key larval sources, so that would be a good target area for further protection, especially if they concentrate or congregate with other highly fecund colonies. And then finally, this provides baseline data for both the classification and management of future vulnerable marine ecosystems. So these are the people I would like to thank, my supervisors, friends, partner, and COBRA funding. And you can scan this QR code for my references and contact information. And that is it.
Okay. Thank you. So congratulations.
I know, sorry.
Congratulations. Nice presentation, really. Very interesting. I have a few questions. I don't know if you can come back on slides of effective fecundity. At some point, you mentioned the size of, you mentioned the number of gametes per polyp. Have you considered the size of the polyp as well?
Okay, that's a really, really good question, actually. Because I can't find the slide. It like got me out of the presentation. Effective fecundity. Yes. Yeah, so that is something that we considered looking at, the size of the polyp, but because our samples were 20 years old, they were not really preserved properly. So a lot of the times when we went through the slicing and staining process, the polyp kind of exploded, for lack of better terms, but there was still like the basal part where you could see the oocytes and spermatocysts at the bottom. So we could still extract some information, but there was no like
measuring the size.
Yeah, there was no measuring the size of the polyps. And it's not a really well-studied species as far as like morphometrics go. I know there have been several studies on like symbionts, but yeah.
And when you were comparing the two species of colonies, so maybe with a few slides, here I cannot not realize that you have a very big sample size difference.
Yes. Yeah.
So what's the reason and how much could be a problem?
It's definitely, this is not like a true comparison that I would publish. It was just an interesting fact because I was given the opportunity to work on a more fresh sample so I could go through the whole like decalcifying process and everything, which I wasn't able to do for the legacy samples because they came preserved in giant wax blocks. But it's also the deep sea, so it's very like opportunistic as far as sampling goes. So they only found one Iridogorgia colony during this cruise, and it's the only one my supervisor had access to. So that's what I got to do. But there is some potential talk about me accessing more in the future, so I can continue to add it to my study.
For you. And maybe the last one, you mentioned that the seamount, the summit of the seamount seems correlated to fecundity. But at the same time, it seems that you find no correlation at all between, so I guess you mentioned that to, let's say it again, you consider possible effect due to nutrient supply here. But at the same time, to the left, there is no connection with productivity and chlorophyll A.
Correct.
So how do you explain that seamount summit depth can be related to fecundity due to the fact that it supplies nutrients and we don't see any effect on productivity?
Sure, yeah. I mean, productivity was found to be an effect in several other reproductive studies. So it was kind of surprising that we didn't see a correlation there, but my best guess is that the productivity metrics are quite stable along the New England seamount chain, and there's not like almost every seamount we looked at had pretty stable conditions, but there was some variability, so maybe it didn't have like a huge impact on reproductive output because they had access to nutrients more consistently, and maybe the current transport was just like stronger, and the like access from the surface to the corals was maybe stronger at these other locations.
So would it be that the seamount summit depth may have an effect on fecundity aside from nutrients?
Yeah, like the seamount summit depth interacts with currents a little bit more strongly. So like seamounts that have their summit lower may not have access to the nutrients up at the surface as much as those that have shallower summits. So it's like, while the nutrients are over the whole seamount chain, the specific hydrodynamics at each location could influence their ability to access the nutrients more.
Next one.
I will just come back to that, but first of all, congratulations for your very nice presentations, and I'm sorry the figures didn't work out,
It's
but
okay.
it's very good and nice. No worries. So was just thinking for what you discussed before about the productivity issue. Like I'm just wondering. I don't know. Is it the chlorophyll A the right way to discuss productivity about the deep sea?
Yeah, it's debated because there's not a lot of food sources. The deep sea is like a food poor region naturally, but I mean a lot of other studies have correlated deep sea reproduction to surface food fluxes, especially for continuously reproducing corals. If they have like known seasonal reproduction, it is tied to the phytoplankton blooms throughout the year. So it's a pretty like semi-known fact at least that chlorophyll A is a factor, but there's also food falls like whales getting to the deep sea and decomposing that also like could be potential food sources, but it's just what my supervisor suggested.
Of course, it's generally also accepted that the common way for that, but because it's more in the surface productivity, also you lose a lot of the organic matter that is in the deeper layer. And therefore, I think also is why you cannot have. Yeah, it happens in many studies. I mean, in fact, that patterns are not shown based only on chlorophyll, but it's just a comment for the discussion.
No, of course, yeah. I suggested we needed to do in situ measurements as well of the food flux, which
I was also wondering about the temperature because also you mentioned environmental applications not related to fecundity. I would expect there is some change, but what about temperature and salinity also? How was that measured?
that was
And isn't that a bit like kind of stable in the deep sea?
yes. Yes, yeah. So the temperature and salinity actually came from the ROVs that collected my samples. So we did actually get like at the collection site data for those. But yeah, again, there were some studies that I had read that had found that temperature does have like a strong influence on reproduction, but it was more of a like contrasting depth range that they tested. And my depth range was over, I believe like 1,000 meters. But some of them were testing like 400 meters versus 2,400 meters, and mine were all pretty much deeper than that. So the conditions were more stable, which is what I suggested would be the reason why there wasn't a correlation with temperature and salinity in my study.
It makes sense. It makes sense. And then the last one, if I may, also just to can you go back to this effective fecundity graph? Also, you discussed just curiosity because you discussed the lower fecundity per polyp, the first one, but I was wondering more on the figure on the plot on the right, that it shows a very big variability. And do you have an explanation of that, or what was that population?
Yeah, so this one was the first colony I started off with. So I actually sampled every single polyp we had access to. So this one had like 3,000 polyps that I sliced versus closer to 20 for these two. So that explains the higher variability over here. I put the numbers of the oocytes assessed above the curves here. So you can see there was like quite a large difference between them. And yeah, there was a lot of immature oocytes and some polyps that were not very reproductive, so you have a congregation down here, but then there were polyps that were like insanely reproductive. It kind of felt random, but if I know some other samples we had that I wasn't able to work on had distinguished between if they were from like the apical portions or the central portions of the coral, which would have been super interesting to look at in this case, which could explain a lot of the variation as well if some of these were from the middle versus the outer ends, which would tend to be more reproductive. But it wasn't categorized like that in 2005, so I don't have that information.
Okay, thank you very much.
Thank you.
Thank you, Amanda. Next presentation. I'm going to just make some questions about the methodology of histology.
Sure.
Did you work? I mean, which is your specific work in the study?
I did everything.
All histology?
Except for collect my own samples. I was not there in 2005. But I did all of the
That was a pity, but but you make all the histology. Any problem?
Oh yes. Well, most of my samples were pretty old, so they were decalcified using Holland's fixative, if you've ever heard of it.
That was my next question. So the classification problem.
Yeah.
Why? Because of
Why?
octocolors, no?
Yeah. No, there
We
was.
use octocolors, we don't decalcify them.
But, for some, well,
Well, I wanted to ask this because for
Yeah,
me it
it's-
was
because we are working also with
Yeah.
not the other.
The
we have
axis
to
is.
classify. So it was
okay.
was the question.
Okay.
Yeah, the axis is still very brittle.
Okay.
It's not the polyps that are there's no like sclerites that cause the problem. It's the axis that causes
Okay,
the
I see.
problem. But because they didn't there was no reasoning for using Holland's fixative, at least from my knowledge, because it's not used in marine biology. It's like human
we use,
gastrointestinal.
EDTA.
Okay, we used like a decalcifier too. It was just like some diluted hydrochloric acid. But yeah, for most of my samples, they weren't decalcified. And so there was a lot of like ripping during the slicing process, which caused the exploded polyp situation. But yeah, the fresh samples that I was able to work with Iridogorgia, doing the decalcification using the hydrochloric acid really helped remove the axis.
Because it's easy to have problems in
Mhm.
the sectioning.
Oh yeah,
For instance. Yeah. Okay.
lots
So
of problems.
there were problems also in. Okay. Good. It's no, it's good. It's good. Yeah, to see that.
Yes,
you're
other
not doing
people
anything
also-
wrong.
Of course, it's important. Any hermaphrodite?
No, not in my colonies.
No.
My lab mate saw one though
Ah,
in Paramaricia,
okay.
not mine.
And apart from with the fecundity and the mature gametes that you observe, which is the control just to see the section is the proper one and the size of the section of the ovocyte is in the middle or not? Do you have any control
Oh,
just
to like
see the maturation if it's
for the
yes?
sorry, I'm
when you
not
analyze-
understanding the
when
question.
you analyze
Mhm.
the mature ovocytes,
Mhm.
how do you know that they are mature and not? They are.
Oh, I based my
It's
classification.
just for the size of the oocyte.
Yeah, it is based on size, but I also based off a lot of literature that had like very intense descriptions of each stage class.
Yeah.
So for these guys, it's just like the very first buds, and then for pre-vitellogenic, they have like a central nucleus, and then vitellogenic is classified by rapid yolk accumulation. So it's like when the yolk gets bigger, and then this guy is like grainy and stained a completely different color, so it's super easy to tell. But yeah, it was mainly based off descriptions from the literature because size differentiation obviously varies by species. So I kind of made size ranges myself for the classifications once I identified them based off of descriptors.
Okay, good. Okay.
So that was very nice. So I just have a quick question. If you could plan new surveys to understand better these issues or to advance knowledge about the species, where would you go? Have you ever looked at a map and say, oh, there are good spots to study this?
Yeah, of course. I would love, for the Metallogorgia specifically, I would love to get a characterization at the other seamounts in the New England seamount chain, especially the three closest to shore because those are the ones that just reopened for the fishing pressures, just to see what that looks like, either before or after that happens, so we can have a bit of a comparison of different conditions. Because I did have a couple of seamounts that were clustered and like the colonies were clustered along the same seamounts, so I would like to see the other ones, but originally my idea for this study was to compare the same species across ocean basins, so I would also like to find Metallogorgia in the Pacific to see if there's any differences there, and vice versa for the Iridogorgia if we could get some from the Atlantic, which are the samples that I'm about to work on, so I'm excited.
And would you use the same methods or would you do something different?
Yeah, for sample collection, we can't really control what they do as far as that goes, but for the staining process, there's actually Maxon's trichrome. I don't know if you guys have heard of that, but it's there's a higher contrast between nucleic material and like cellular other things. So it's a bit easier to tell between the gametes versus the rest of the material in the polyp. But I'm also so used to everything being purple now that maybe I would keep the same methodology. So.
Okay, thanks a lot.
Good, thank you.
Thank you.
Just lower the font a bit because I think that's the problem. I think it's like going into the red. This one, right?
Yeah. Okay, now we have daily with this influence of temperature and food availability on the survival, growth, and metabolic rates of Atlantic blue tuna larvae. Okay, floor is yours. 15 minutes, and 2 minutes are left. I stand up.
Perfect. Well, hello. My name is Kaylee, and today I will be presenting to you my thesis entitled "The Influence of Temperature and Food Availability on the Vital Rates of Atlantic Bluefin Tuna Larvae." Atlantic bluefin tuna are highly migratory pelagic predators and are one of the most desired fish in the world. One of the main spawning sites is in the Mediterranean Sea, and as you can see from this year-long migration of attack individual, they swim into the Mediterranean in the summer to spawn, and then return to foraging sites across the North Atlantic for the rest of the year. The larval stage is characterized by fast development, with critical organs fully functional by about 20 days post-hatch. So one of the main spawning sites, as I mentioned, is in the Mediterranean, and it's a warm, oligotrophic spawning and nursery site, but as global climate change progresses, temperatures are getting even warmer, and this is causing some community shifts. In zooplankton specifically, they're seeing a community shift from more of a copepod dominance to more of a meroplankton dominance, and this causes the predators of these zooplankton, such as tuna larvae, which feed mainly on copepods, to either have less food available or to have to switch to different food, which may have different nutrient profiles. So, warming seas and high temperatures are leading to lower food availability for some species. So, tuna are asynchronous spawners. They spawn over a period, and the time at which they spawn may have effects on the larvae. So larvae need to hatch into a high food environment in order to fuel their fast development that I mentioned. So a larvae that is spawned early in the season has more favorable temperatures and access to more sufficient food, whereas larvae spawned later in the season is in higher temperatures, which will increase their energetic demand, and so the food available may not be sufficient to fuel. And also, as I mentioned from the community shifts, they may have lower food available in the first place. So first, we ran a preliminary study in order to test the diet and also to establish a metabolic rate coefficient. So we investigated the effect of 2 different feeds on the same response variables as the present study, which were the vital rates of survival, growth, and metabolic rate. And so as I mentioned, tuna larvae feed mainly on copepods in the wild, but the copepods can be quite difficult to rear in aquaculture, so aquaculture typically feeds rotifers instead. However, rotifers are known to have lower protein as well as different fatty acid ratios. So with this study, we verified that copepods are the ideal first feed for tuna larvae, highlighted by a higher survival as well as a more uniform growth within the cohort. So we use copepod monopoly for the feeding in the present study. We are also able to calculate a strong allometric metabolic rate equation, which is only the second equation calculated for the Atlantic bluefin tuna larvae. And this gave us a slope or a metabolic scaling coefficient of 1.13, which we then used for the calculations and the comparisons in the present study. So now to the present study. This study aimed to test the effects of temperature and feeding on the vital rates of Atlantic bluefin tuna larvae, specifically the survival, the growth, and the metabolic rate, which are all regularly used variables in biological fisheries models. So we tested the combination of these stressors in a factorial design. So overall, we expected that higher temperatures will have stronger effects on the vital rates with increased growth and increased metabolic rate. We also expected that low food availability will decrease or suppress the vital rates, but also that this decrease will be more obvious at the high temperatures than the low temperatures. Specifically for warming conditions, as I mentioned, where high temperature and low food availability, we expected low survival, which is driven by energetic deficiencies. High growth, but not as high as if there's both high temperature and high food availability. This disparity comes from, again, the increased energetic demand from high temperatures, but then an inability to fuel it all the way. And then lastly, a high metabolic rate in conjunction with our expectations of increased cellular function, increased energetic demand, increased growth. So the eggs for this experiment were collected from a spawning event of Atlantic bluefin tuna broodstock off the coast of Spain, and then were then transported to the IEO experimental facilities. Once the eggs hatched, they were moved into their treatment conditions, which were designed based on a 2-factorial design of 2 food levels and 2 temperature levels. The low temperature level was 22 degrees Celsius, which is a normal spawning temperature in the Mediterranean, and then the high temperature was 26 degrees Celsius, which was an extreme at the time that this experiment was run. And then the high food treatment was 10 copepod noculi per milliliter, and the low food was just 1 copepod noculi. The low temperature high food treatment here is meant to represent a normal spawning condition in the Mediterranean, and then the high temperature low food is meant to represent a warming or a heat wave condition. This experiment was run for 11 days for the high temperature treatments and 14 days for the low temperature treatments. The survival was measured in terms of total percent larvae that survived to 11 days post-hatch. All treatments were sampled for growth at 11 days post-hatch, and this was for the dry weight, which was taken by freezing it and then taking the lyophilized dry weight. And the specific growth rate, which is the change in dry weight per day, was then calculated from our dry weight. The metabolic rate was measured at 11 days post-hatch for the high temperature larvae and 14 days for the low temperature larvae. And this was to try to ensure that all larvae were about the same size when the metabolic rate was taken, as the developmental stage may have an impact on their metabolic rate. And so the researchers noticed that the larvae in the low temperatures were growing slower, and so they let them grow for a few more days. The metabolic rate was taken in terms of routine metabolic rate, which allowed the larvae to swim within their vials. And so the larvae were moved from their treatments into the closed system respirometers, and the oxygen saturation was measured over the course of 60 minutes. Since the larvae were not all the same size, the mass-specific routine metabolic rate was calculated in order to standardize the metabolic rate per size. So looking at some results now. Here we have the total percent survival of each tank per treatment, and each point represents a tank. And as you can see, there's very high variability within each treatment for the tank survival. This was an average survival across the treatments of about 3%, and no significant differences between the treatments. Typically, there is very low survival of Atlantic bluefin tuna larvae in aquaculture, with an average of about 3 to 4% survival to 20 days post-hatch, so our average survival does fit within this. But it's important to note that the low survival of the larvae is one of the main issues in tuna aquaculture, and this is particularly driven by the high sensitivity of larvae to any external factors such as light or microbial presence. So the growth results did follow our expectations, and as you can see here from the specific growth rate, where each point represents a larvae and the lines represent the average specific growth rate per treatment. Each treatment was significantly different, but you can see here that our high temperature treatments, both of them, do have much higher growth. Increased temperature is known to increase growth rates by increasing the cellular functions from reaction rates all the way up through cell growth. So the fact that our high temperature treatments were growing much faster does make sense. When we look at the food availability, we see a larger difference between the feeding availability in the high temperatures than in the low temperatures, as you can see from this wider gap between our temperature treatments, between the food treatments at the higher temperatures, which shows that the feeding treatment was a limiting factor in the high treatments. So at its simplest, the energetic balance for the energy required for growth can be modeled as the consumed energy minus the metabolic energy. So the more a larvae consumes, the more it will have to allocate for growth. However, if a larvae is not consuming enough to fuel past its basic cellular demand, then it will not be able to grow as much. So what we're likely seeing here is that as the energetic demand increases in the high temperatures, the larvae are not eating enough in the low food treatment, and so there's this greater disparity and this greater gap in the specific growth rate in these. So there are multiple implications for low or stunted growth in larvae. It's been said that the mortality of larval fish is driven by starvation and predation, as a larvae needs to be able to grow quickly to outperform predators. Specifically, as a larvae grows, the swimming ability gets stronger, and also its visual sense develops in order to see prey and predators. So a larvae is less vulnerable to predators. But if a larvae is underfed or grows more slowly, then it is smaller for a longer period of time. And so it may be weaker, have a weaker swimming ability, so it can't escape predators as quickly, or it may spend more time foraging for the few prey in the water column, and so it's more exposed to predators. So smaller larvae are more vulnerable for a longer period of time. So we compared the metabolic rate between treatments using the mouse-specific metabolic rate. And as you can see here, there was very wide variation within each treatment and no significant differences between the treatments. Looking specifically at our warming conditions again, which is here in the high temperature, low food, our expectations kind of created a paradox where we expected an increased metabolic rate in high temperatures, but then also a suppressed metabolic rate in low food, as some other studies in adult fish have seen a suppressed metabolic rate. So even with this paradox, we do see a consistent metabolic rate in all treatments, which points to a stability of metabolic rate even with environmental stress and stressors. Another study has done a similar experiment and seen a compensation ability in adult sardines as they had a lower maintenance mitochondrial cost. And so it's possibly that our larvae are compensating or have a resilience in some similar way. However, that study was on adult fish, not larval fish, which are very different. So the Q10 is the factor of increase of metabolic rate or other biological rates between 10 degrees Celsius. And this is a commonly used value in different models in order to predict the effect of warming on biological rates. So we calculated the Q10s here, and our calculated Q10s are here in the orange box. So looking at the high feeding treatment, which removes the feeding stressors and only focuses on the effect of temperature, we see a Q10 of 1.07. And this means that there's virtually no change in the metabolic rate with an increase in temperature, which again points to a resiliency or a possible compensation of the larvae to retain a constant metabolic rate, even in stress. However, when we add the low feeding back in, so we have both stressors, we see a Q10 of 1.39, which shows an increased sensitivity of the larvae when there's a combination of stressors. These are the first Q10s calculated for Atlantic bluefin tuna larvae. And so previous models have assumed a Q10 for this species and for these larvae, but this assumed Q10 is typically 2, which has been assumed based on similar species such as the Atlantic mackerel, which has a Q10 of 1.7. So this study very much counters these previous assumptions and indicates that we may have been over-assuming our Q10 in models and points to the need to improve models and also biological amounts to have better predictions. So the metabolic rate of larvae is notoriously difficult to measure, but it needs to be improved in order to do better research, have better knowledge, and be able to compare against studies. So one thing is that we need to develop standardized methods. There's a wide range of methods used from calorimetry to respirometry. So we need to make sure that we can use the same methods so that we can actually compare the studies and the metabolic rates. We also need to agree on a metabolic rate measurement proxy. A lot of studies will use a standard metabolic rate, which uses anesthesia to remove any activity of the larvae, but anesthesia may have an impact on the larval health and then may also impact the metabolic rate. Whereas we use routine metabolic rate, which allows the larvae to swim within the respirometer. As I mentioned, larvae are very sensitive, and so they have low aquaculture survival, so low ability to have in experimental tanks, and also very limited handling to be able to move them from the tanks to the respirometers. And so it needs to have better handling ability or better respirometry ability in order to cause the least amount of stress and the least amount of effect on the metabolic rate. So as we can see, as we have seen from this, the combination of stressors amplifies the negative effects on the morphology in the Q10. And this is likely driven by food availability as a limiting factor at higher temperatures. We also saw a resiliency in the routine metabolic rate in temperature stress. This study really emphasizes the need to consider biological effects and combinatorial effects when creating models of marine populations. Many fisheries managements use models to inform how you should manage the fishery, but many climate change models are focused on the distribution or the movement of species or fail to consider overlapping effects. For example, if we created a model using the specific growth rate from our high temperature, high food larvae, we would expect fast growth of the larvae, big tuna, good population. But then in reality, if the larvae are also faced with prey shortages driven by these warm temperatures, then the larvae would actually grow much slower and there may be mortality increases as well, and this would cause our model to be incorrect and possibly mismanage a fishery in an extreme case. Adding specific physiological effects will increase the accuracy and the flexibility of models, help us to better predict the abundance and the health of tuna stocks, and also help to inform fisheries management. So thank you so much, and thank you so much to my advisors and my team, and also the experimental team at IEO for their work.
Okay, thank you. That was really nice. So I just have a few questions. So based on your results, would you say that this species larvae under these conditions is pretty much operating under its optimal thermal interval?
Like under our experimental conditions. Looking at some of the results, let me just focus, for example. I mean, we'd see with the larvae that basically they have, of course, an extreme temperature, but really with larvae, the higher the temperature is, the better they do. I mean, it's kind of like a chicken sitting on an egg to warm up the egg and help it develop. So development is better in warmth, and high food as well is what we expect. So it's interesting because the actual environmental one that we expect, the low temperature high food, is typically not as ideal when looking at growth and stuff. They grow much lower, but also there's this trade-off in the environment with the food versus the temperature. So I think if there was high food all the time, we would expect to see the larvae spawn much later in the higher temperatures. But there's really this trade-off that they have to make when they're spawning in order to have both sufficient food and favorable temperatures.
So let's say that you would like to have some idea of temperature where thermal stress would start. What would you do to complement your study?
Yeah, I mean, we could do another study on just trying to continue to increase the temperature. Again, because of this, in the environment, this interaction between warming and prey, because of the whole trophic chain and everything, we may not have the right experimental conditions, because of course in experiments in the lab you have to take it one variable at a time. So I think it would be interesting to just stay with one food source and just keep increasing the temperature. Again, look at the metabolic rate and see when they're having stress, and then look at the survival as well to see if there's any changes there. But we could also continue to do something with the food, but that may come later after we find just the thermal maximum at one normal food.
And did you look at the temperatures that are currently occurring every summer and to see if your species is under optimal conditions or if it's maybe higher?
Yeah, so that's definitely one of the limitations of this study. This experiment was ran in 2021, and so at the time of that experiment, the extreme was 26 degrees. Now we're seeing increases of up to and more than 30 degrees. So I would love to run this again and see now what's happening. It's likely that other experiments in the literature still haven't found a thermal maximum for the species, so it's possible that now it could be similar, but of course the zooplankton are also changing their phenology, so we might have more of an extreme in the high temperature low food. So
Do you know if the high temperatures coincide with there being larvae at the fish at larval stage in that area?
Yeah, it's very similar spawning patterns, because I mean they're actually on a boat right now. I'm not sure if they're watching. My supervisors are collecting larval surveys currently, and so far up to this point, the larvae are still spawning kind of at the same abundance and same areas, even with the higher temperatures. But that's just right when they're spawning. We're not really seeing the same development over a period of time. It's just one time point.
And when you, as you did some respirometry, correct? Did you do it individually for each larvae?
Yes, each one was in its own little, like 4 milliliter.
But you had them in tanks with several larvae, correct?
Yeah, so they were grown up with cellular larvae, and then they were moved to individual respirometry vials in a water bath, and then they're multiple measured kind of at the same time.
And when you observed them in the big tanks, did you notice if they had some shoaling behavior where they move in groups?
So I wasn't actually there for this, but currently they're also running some experiments on the schooling behaviors, and they're seeing it being developed more in the juvenile stage, so 30 days plus.
Okay, so
Yeah.
they show after the larval stage.
Yeah, so
Okay.
larval stage is still very individual, just swimming as they can, but yeah.
Okay. Thank you.
Thank you, Gail. nice to see you here again.
It's nice to see you.
You know, probably you know that the people that is working really nice with the topic when came here with the trousers related to the topic.
I mean, I think I obviously have a little bit of a passion.
So, that's nice. That's really good because it's something that also is part of the master, I guess. So really cool. Apart of that,
Please.
26 heat wave.
Mhm.
Neither in Basque Country.
It's progressing fast. Yeah.
Because it's not. So that was also my question. I mean,
Yeah.
because
Again,
26
I wasn't the one who designed this experiment or ran it, so if I had had it, I would have had maybe more temperatures if possible, and also higher temperatures.
But it doesn't have to be a heat wave, it could be other conditions. I mean, because even if it's not a heat wave, it's going to be the regular temperature. Okay, we can see that the high temperature could have some effects on the larvae. But what about the copepods?
Yeah, so with copepods, with the shift in the communities, it's really they're shifting their phenology, so the copepods are spawning earlier. But also in the Mediterranean, it's an oligotrophic sea, so there tends to be low food for most of the time. So larvae are.
But I mean in the experiment because
They were feeding them every day, so they're putting these copepods into the tank every day. So even in the treatments,
but the copepods was at the same temperature
Mhm.
of the treatment group.
like rearing the copepods,
They grow at 26 degrees and 22.
I'm not sure. I'm sorry. I think it definitely would make a difference if they were shocked into.
Because they are going to accumulate different components, they are going to
Yeah.
have different organic,
Yeah.
I mean, lipids and distribution.
From
And
what,
also probably the behavior of the copepods in the water, cooling the food.
absolutely. From what I've heard, even about, I went to the aquaculture center in Murcia, and they had difficulties even rearing copepods in the first place. So I think that trying to rear them in different experimental conditions, different temperatures, would probably be an extra stress. But yeah, it's definitely an effect of shocking them into warmer temperatures or cooler temperatures. And how they
Because
grow,
here we have
their
an
nutrients.
copepods, and it's always telling us, we have this strain of copepods, but it's not adjusted perfectly well to this temperature, so.
There's always another level.
What about the ethic permissions and these kind of things for these experiments?
I think we weren't allowed to starve the larvae, so that's why they still have one copepod per milliliter. If we really want an extreme, we could probably drop that even more, but not ethical. So just trying to stay ethical. They did the ethic permissions and everything beforehand, but it's also a larval species, so I believe.
Sometimes it's good to use
To put this in here.
I know the people and I know that they are working perfectly well, but
It's in the written
well, that is
manuscript if you ever
important.
get to read that. Yeah, the presentation, it would be good to definitely next time, remember.
So finally, which is your thesis?
My thesis is on the heat wave one, but hopefully we'll.
I mean the
Oh.
thesis. Thesis means conclusion.
Okay.
You have an
I wrote two
hypothesis.
different papers, so that's why I thought you were trying to guess which one was actually the thesis. Yeah. So basically, the big thing is that just this larvae might actually be quite resilient in stress, which is quite interesting because larvae are so sensitive, but they might be doing quite well. But then also that I'm very passionate about both biology and fishery science, so trying to better inform models. So what I'm really hoping is that kind of the specific growth rates as well as the Q10s from this study will be able to be used in models in order to better model and also help for future predictions of fisheries. So basically, we have more resilience than we thought we did, but also we need better numbers. And since larvae are so hard to measure, this is kind of one of the first studies on the metabolic rate and the growth of the larvae at these conditions. So just informing the people in the science and the policy is my hope for all of this.
No, thank you.
And really, very nice presentation. Thank you, and yeah, very well. Also, I enjoyed the discussions here. I'm the exposure conditions, of course, it comes first. The food will also be impacted, so you're gonna be feeding on a less quality food. I was also thinking about that, but I guess it's pretty clear that I think the copycats were not treated beforehand. But do you know what kind of copycats they use? Is like a single species or mixed culture or
I
what
believe
was
it was
it?
all Arcancia. It's all one species, all at the Napoli stage. And then I know, like, as we go through because this was only until 2 weeks, but then at about 15 days, then they start changing the feeding to larger feeds and full copepods, not just the Napoli.
Okay. And what was the low and high density? I did not understand this very well.
Sorry, I didn't put numbers on this little thing, but the high density was 10 copepod Napoli per milliliter, and then the low was just one. Typical aquaculture uses at least 5 copepod Napoli per milliliter, so this is quite ad libitum and feeding them quite well. So they should
Just to make sure I understood correctly about the one, you know. That looked a bit low, but okay, no worries.
as low as we could go.
I would, I was thinking about that, like, because you also mentioned like high temperature and low food availability, so the worst combination. But there can be like the scenario of high food availability and high temperature. So there can be high food availability, but the food quality itself might be not as
Yes.
good due to the high temperature. Of course, this another study to do that. But what would you consider if you are expanding this study and you want to measure also the quality?
Yeah, I think there could be definitely some really good lipid profiles done on the food because highly migratory fish, tuna, they mostly need high protein and certain lipids in order to fuel their fat storage and all their swimming. So most important is protein and lipids. And so doing some studies and some lipid analysis on the copepods, maybe comparing the low and high temperatures would be really interesting. There's been a lot of studies on, that's why we ran the preliminary study, because there's been a lot of studies that have compared the copepods to the rotifers. So kind of replicating those, but just copepods at different temperatures or different environments would be really interesting as well. And then if we need, for other studies, if we want to run this again, if we do notice differences in lipids, in aquaculture, rotifers are generally enriched with protein and with fatty acids. So if we have to enrich copepods in order to try to get to the same stage so we can see similar results is possible. But yeah, definitely just doing some lipid and protein analysis would be really interesting.
Yeah, that's very good. Yeah, I was also had my critics about the temperature and this, but of course not your fault. It is maybe this clear.
I agree with those.
And but just a small detail about the respiration measurements. You did very individual, right? You
Yes.
corrected this for biomass. Is that right? Or
I corrected it for the mass for each larvae because again, even when we tried to let some of them grow for longer to create different, the same size, there was still quite a size distribution within that. So tried to standardize.
yeah, that's very important because sometimes I'm working on this, and sometimes in the studies it's reported per individual, and sometimes it's reported per biomass, and it's like
So we
also
have
a different, completely different measurement. So it's good that you took it into account. Yeah, it's fine for me. Thank you very much. Thank you.
Well, congratulations. It's really clear and been able to address at the same time some technical aspects and how to do that with little fish in little jars to some ecological aspects. I will go to this ecological aspect that I find very interesting. So it's really clear that here it seems the larvae can compensate this warming up if they have food, and that's nice. And it can connect to a lot of this big hypothesis: fisheries bigger is better. Also the mismatch. So how did you started tackling the aspect of this bigger is better aspect? What can you say of this realization here? What your results towards this big ecological hypothesis?
Yeah, so I was trying to relate it to a little bit to the mismatch hypothesis, as you mentioned, but oligotrophic systems, not quite a mismatch thing because there's quite similar zooplankton throughout the, it's not as much of a peak
Okay.
as there may be in other ecosystems. It's kind of just little lumps. So really the issue is not just that, yes, there are community shifts and there's changes with that, but also the big thing is the temperature driving the energetic demand up. So, but I'm
And
sure.
so for this aspect of growing faster or less fast, because you've got here impact on the growth rate difference, according so to the food availability as well. So how much can be a difference to evade faster this predation?
Yeah, I mean, if they're not getting nutrients, if they're not able to grow, then definitely more exposure to predators because it is a big thing with they develop within 20 days to be able to outperform the predators.
So 20 days, okay.
20 days
And
is
how
really
long do the larvae stay without having to eat, or to just focus on the yolk?
They hatch within about 1 to 2, I think 48 hours after fertilization. So it's a very quick stage all the way up. So they hatch quite quickly, and then the mouth opening is about 2 days after hatching, and then after 3 days they actually start feeding on actual food. So I think there's about 2 days where they.
And do we know, really technical question maybe to finish, but do we know also at what point they become kind of active swimmers and deciders?
They're active. I mean, not floating.
Okay.
With like,
Just nectar.
like pretty much nectar as soon as they get up. Active swimming, I'm not sure the exact time that it occurs, but it is within that 20 days of, and that's when all the critical organs are fully functioning, so heart, digestive tract, and everything. But swimming is one of the first things that they do because they need to prioritize being able to consume and being able to swim. So definitely digestive tract and fins are like the first things that they really develop. Also the eyes are super important so that they can actually see.
Yeah. And I also saw in your preliminary study you had this 1 versus 2% survival with the copepod versus rotifers. Okay, so 98% of. And you say that it's 3 to 4% survival in aquaculture.
Yeah.
But in aquaculture, we can imagine that they're not totally controlled environments. Here you were in a lab with controlled environments.
Yeah.
So what's the main cause of mortality?
Mortality in aquaculture, it's mostly, there's a lot of things. There's the collision into walls. They get stuck on the surface a lot because they're so light that they go up to feed and then they just get stuck on the surface of the water. But also there's a sinking issue, so it's as everything is kind of developing, it's kind of chaos. So their swim bladders can get messed up and again stuck, or sinking issue as well. So there's a lot of research being done specifically in Japan on trying to just get the larvae to survive. There's other fish species, I think sea bream.
But sorry, but specifically here for your study,
Oh,
because for your study you were in a controlled environment of the lab.
Yes, I'm not sure what our, I wasn't there to run it, unfortunately. I don't know what their main causes of mortality were, but again, to aquaculture, it's usually collisions or sinking or getting stuck to the surface, and that's what other aquaculture or other studies have seen mostly in there. But I don't think they took notes on exactly how they were dying.
Thank you.
Thank you.
It's okay, you can start, I'll keep the time for you. If he's going to the bathroom, I'll keep the time for you. You can come over. Okay, they're ready in 15 minutes, and I'll let you know when there's 2 minutes left.
Good afternoon, everyone. My name is Devani, and I will be telling you about my thesis topic, where I created protocols for microscopy-based studies looking at the morphology and organelle distribution of a single-celled, a giant unicellular multinucleate algae called Caulerpa. A mouthful. So Caulerpa is Siphonis. This just means that it's one giant cell that has no cross walls on the inside like normal plants do. So that means that it has filaments where the cytoplasm can move freely throughout the entirety of the plant. What also makes it interesting is that despite it being one single cell, it can grow into different parts. So it can grow into the front, which is like a leaf, into a stolon, and then also into a root-like system that are called rhizoids. So there's a lot of studies that have been done on optimizing clearing protocols and staining for plants, but not really for algae that much, and that's mainly because it's really difficult for to get rid of the pigmentation. It's very autofluorescent, and usually they have very thick samples to work with, which makes it optically difficult to image. So using some plant information, looking at the transparency, the clearing methods that they use to get the best transparency, to limit or increase or preserve the fluorescence, and then also to look at the best methods to keep the tissue integrity. So the aims for this study was for me to create a workflow where I can do qualitative imaging of Caularpa species, not just of one, but hopefully of all of them, in order to, with the main focus on keeping the structure of the organism, but also being able to see into the organism properly and using same compatibility with the clearing methods as well. Secondly, I wanted to compare all of these methods. So looking at the different clearing methods, the different stains, and the different microscopy approaches, I wanted to see which combinations of those worked the best for the most information on the internal structure. And lastly, I wanted to create a practical qualitative analysis workflow in order to assess the background interference and other imaging possibilities to see the most of its biological nature. So for research questions, we wanted to find out which of the sample preparation clearing methods were the best, that focused mainly on the structural integrity, but also keeping the internal visibility. And then which stain combinations worked the best that we could find the most information from. We also wanted to know which microscopy platform would work the best specifically with these thick samples. And then which of these together with the workflow showed us the most information internally. This is just the workflow that I followed. We sampled two different Caularpa species. One was from Portugal, so it was a local species, and the other one was from the Caribbean. We did fixation, clearing using two different methods and 4 different stains. We were looking at it under 4 different types of microscopes, and also using the imaging software of each of those microscopes to get the image processing. And then lastly, this is an ongoing study. I have not submitted my thesis yet, so I used ImageJ for the preliminary results, and I will be using R for some quantitative results later on. These are the microscopes that I used. I started with a very basic Motic upright microscope, which is great for 2D imaging, and just a quick overview. Then I went to the AxioZoom stereoscope, which is great because it has fluorescence, so you can see, you get a really good overview of the whole sample. But for more in-depth information, I went to the Leica, which also has autofluorescence, which you can do 3D imaging. That's really great. But unfortunately, with that one, you needed really thin samples. So I went to the light sheet microscopy, which gave me the opportunity of working with thick samples, but also doing 3D imaging and fluorescence. This is just a quick clearing process. We fixated all of the samples in 4% paraformaldehyde. We did depigmentation, permeabilization, and for some of the thinner samples, we embedded them in low melting agarose for easier imaging later on. And then after that, we went into the 4 stainings, which was propylium iodide and nuclear green for nuclei, and phthalate 488 in Texas red, which is good for filaments visualization. After that, we put them in the two different curing methods. I did cubic and BAP. Cubic is great, it's aqueous and it's non-toxic. It also has a great index, reflective index for microscopy. And BAP, although it's a little bit more toxic, you use methanol for the curing process, and unfortunately, sometimes that does lead to some shrinkage. However, it is great for removing autofluorescence. It's better than cubic at removing autofluorescence. But then for the imaging afterwards, we put it in ethyl cinnamate, which was less toxic, with a much better refractive index for imaging. We compared controls without any stains to see if the curing protocols worked, and they clearly did. So we were able to see past the surface of the plant nicely into it. This is the stolons, and these are the fronds. So here we can see some information coming out on the inside of the plant, which we have never been able to image before. Here we did single stains, focusing on, unfortunately, one of my samples were so cleared that I lost them. But we can see with cubic in the stolons, it works really well with pretty much any one of the stains. You can see quite a bit of information. However, with the BAP, we were able to see with nuclear green really intense filament information, which was really cool. Then we put the two stains together. We wanted to combine stains that had different wavelengths to see if we could see more information, if it made a difference at all. And so we combined stains that were, one was on the red wavelength, one was on the green wavelength, and we always compared stains that one was for nuclei and one was for filaments. So here we can see, again, cubic works well with all of the stains in the stolon, and nuclear green and Texas red was really good at BAP for the leaves. So overall, both of the curing solutions worked really well. We were able to see inside the plant very nicely. We were able to combine the stains, and that all got together. The best solution for this was the double stain. However, there were definitely some uncertainties based off of the curing method in combination with which tissue you were looking at, as well as which stains you were looking at. But we also found that light sheet microscopy was the best use for this. It gave us the best 3D images and the most information structurally. So during my studies, I was able to develop the advanced imaging pipeline for really thick samples of Caularpa, which had never been done before. I was able to integrate clearing with fluorescent staining and 3D light sheet microscopy, also never done before. And I was able to generate 3D images to better see the insides and the structure of these Caularpa, which you'll see on the next slide is really cool. It looks very nice. This is what I was talking about. So there's a lot that still needs to be done with this. We have a lot of challenges. We had some microscopy technical issues. We had issues with mounting because of the thick samples. We also wanted to keep it economic, so we didn't want to buy new stainings or new clearing solutions, so we stuck to the ones that we had in the lab. So we would like to elaborate a little bit more on that. And then, unfortunately, I was also not able to do any replication on this, so there's a lot of information that might be missing. Here, I don't know if it will play, but that is the 3D imaging that we were able to generate with the light sheet microscopy. So for the next steps, as I said, testing more curing methods, other curing methods that haven't been tested before, using different stains as well. That's the next steps. We also want to see if we can optimize mounting on different imaging platforms, not just on light sheet. We definitely need to replicate the workflow to make sure we have the right information. And because the study is not done yet, I would also like to go into some quantitative information on the filaments inside of the Caulerpa. And then my favorite thing right now, which this microscope only arrived after I had left, but this is an uncleared, unstained sample of Caulerpa, which gave us the same information that the light sheet gave us without any of the clearing or time spent. So this would be the next step. They just got this microscope in, so I wasn't able to work with it, unfortunately. But yeah, I think we could get really interesting information from that, and then also doing live imaging to look at the cytoplasm flow now that we can see inside the samples. I would like to thank my supervisors, Dr. Teresa and Ashwin, for all their help, Lexi and Sandra in the lab that helped me, Prof. Rui that helped with spectral photometry, and Dr. Karila that trained me in all of my microscopy. And then financially, this project was supported by Erasmus Mundus and his team. These are references if you're interested. Thank you so much. Any questions?
Okay, I'll come back to me. Okay.
Yeah.
Congratulations, Dominique.
Thank you.
I have maybe two questions. Very interesting knowing so Caulerpa, this is particular to have this big, as you say, organism with a lot of nuclei inside.
Yes.
No separations, but do you know how these nuclei are distributed in the cytoplasm?
That's what we were hoping to find out with this. That's why we did the staining for the nuclei. But unfortunately, when we did the imaging, we were not able to use the imaging platforms, but we could do like 20x or 40x. So with the light sheet, we could only do the 5x. And I feel like that was not enough of a zoom in to see the distribution of the... So we did stain for it. And in some of the images, you can see some marks, but we don't really know if that's nuclei yet. So the next steps would be to find a way to do imaging deeper in.
What's your guess, or with literature, what has been observed? Are there more
The-
or?
So what I read in one of the papers was that they say within the cytoplasm, the chloroplasts kind of lie close to the surface. So my guess would be that it's distributed throughout. Maybe not as much in the rhizomes because that goes into the ground, but it would be throughout the plant. Say again?
So you're talking about the chloroplasts?
Yes, the chloroplasts, but also the nuclei, because the chloroplasts are against the kind of surface.
Yeah.
I'm assuming that it's distributed throughout, but it doesn't necessarily have to be against the surface because of all of the chloroplasts.
Okay.
But I have not been able to find information on that.
But still, you said you have been able to see inside the plant very nicely, maybe
Yes.
for the first time.
Yes.
So do you know why haven't we been able to image Caulerpa that way before? What was the problem? Is it the fitness?
It's
Or
the
what were
thickness.
the obstacles that you actually...
I think it had to do mostly with, because algae in general is so hard to depigment, to get rid of all of the pigmentation because it's so autofluorescent and everything. I know that there are studies being done on the clearing of algae, but it's usually microalgae, not so much macroalgae. So there's not a lot of research on that. And then for this, I know when we tried other microscopy techniques without any clearing or staining, the pigment was so bad you could only see the surface, and then no matter what you do, you can't really see deeper into it, so it doesn't give you any information. So you have to do the clearing to see inside of it. And then you definitely need a microscope that has the ability of doing like 3D Z-stacks with fluorescence specifically. I don't know if that's
Talking of fluorescence, you're mentioning two wavelengths.
Yes. I actually have
Why choosing these two wavelengths, and do you stain so the DNA with something? What was the wavelength you've chosen? fluorescence?
So the main reason why we chose these 4 stains is because we had them on hand. We were looking at other DNA stains as well, but that would have to be ordered and everything, so we tried to keep this as economical as possible. And the reason we chose these for Texas red and nuclear green. So the nuclear green emits in the blue or green wavelengths, and the Texas red is in the red, which means they don't overlap. And the second they overlap, you don't really know what information you're getting. So you're not sure, is this nuclei, is this part of the filaments? So we were trying to keep those lanes separate just for clarity.
Thank you very much for a very nice presentation. Actually, I had the same question here about the fluorescence and the I was also wondering about that. And also, like, yeah, go back to the methodology. I mean, it's not very easy to, definitely not easy at all, to yeah, distinguish and find what is the right stain or whatever works sometimes, right?
Yeah.
But what was for you like the most difficult technical part? Like,
Oh, I
for the
think-
cost, let's say that you can try and limit things, let's say. But what was the most difficult part technically?
For me, originally, doing the first samples and doing the depigmentation and clearing, it didn't work, and I couldn't figure out why, because I had never worked with algae before. I'd never done imaging before either, and so the person that I was working with had done a lot of clearing on other organisms, but not on algae either. So we were kind of trying to figure it out, but then I was able to speak to someone who had been doing clearing on some algae, on the microalgae, and she gave me great advice, so then it became a lot easier. But up until that point, not knowing what I'm doing and not being able to figure out what I did wrong for the experiment not to work was, that was tricky.
And it's also the mounting is also difficult, like to make it right also.
Yes.
Make sure you're using the right staining and so
Yes, I did a lot of research, and we looked at a lot of different stains, and there were a few weeks where I went back to my supervisor and I said, okay, this is the papers that I found, this is the information that I found, and then we tried to work from there. But yeah.
So do you have other alternatives in mind, like if you are continuing this research that you would like to try, if you had
Yeah,
resources?
for clearing, there are some papers on ClearC, which works really well with plant material. So I was hoping that if I continue, I would like to look into that. It seems to be a very mild curing, so it's not the same as BAP, it's not as toxic, and it's very soft on the plant tissue, basically. And it works with a lot of different stains as well, which is perfect. And then for stainings, there was one Huscht, I can't remember exactly what it was, but it was also part of a DNA stain that I would like to see what information we would be able to get from that.
Is that a possibility you can have, let's say, like a false positive result with the fluorescence, like this staining
Yeah.
and so on, that can be interpreted wrongly?
It could, definitely, and that's where the problem lies, is because I didn't do any replications on this. I didn't have the time to do replications. I don't know exactly, when we're looking at the images, are we seeing the stains for sure, or was it still part of the autofluorescence, or what are we seeing exactly? So yes, definitely, there can be some, yeah.
We can only try for better.
Yes.
Just by curiosity, what was the last, the microscope? You said they had a new microscope in the last picture you showed us. What was the
So
type of
this
microscope?
This is called a confocal microscope, and it's amazing because it gives you all of the information that you would need multiple machines to get. So it gives you the spectral information. You can even divide, you can remove the stains from the fluorescence based off of how long the light lasts, basically. So there's like a death rate for each stain, and then you can remove them. You can see what is actually the stain, what is autofluorescence. So it seems really interesting. Yeah.
I hope you have the chance to use it and work with it.
Me too. Thank you.
Thank you.
Thank you very much. I think that this kind of works are really interesting because it's something we expect, but I mean, research on new methods, new is so hard, and I like a lot the sentence you say, I not knowing what I was doing
Yes.
in some moments, and it's similar happens to me every day.
That's very good for us to know. Starting out,
No,
making
I
all
mean,
mistakes.
this is the challenge, and it is really nice because you try to find the solution. So and I
Thank
think it's
you.
really nice. Well, I'm also working in histology, and I know Teresa recently. I meet Teresa,
Ah,
and we
she's
have
amazing.
we are trying to do some works, and I know how amazing the tools they have are. I mean,
Yes.
this is incredible.
yes.
And this is the question. This microscope, do you know the cost?
For a session? Or
This
the
one,
microscope?
that one
Oh.
you have. I don't care.
I unfortunately don't, but I can definitely put you in contact with a person who does know. So Karina.
Oh, I know more or less.
Okay. I unfortunately
I-
don't.
I
No,
don't- I-
she was also able to get it for a very good deal. I know that. So if you want to speak to somebody who knows how to do that, I can put you in contact.
No,
Okay.
I have some budget and I cannot pay that.
Yeah. It's a lot.
But yes, it's so expensive and it's so important to understand that these machines are quite expensive. I mean, you know,
And even using them, even if I do get to do my PhD, I would have to have a lot of funding just to be able to use it. And the learning curve is insane. I was there when they did this image, and it's not the same as with other microscopes where you can have a 2-hour session of training and then more or less you can start playing around. You will have a week or two
Yeah.
minimum to try and understand everything. So it's very, very intensive.
sometimes for me it's even comparable with the electron microscope sometimes. So
I've not used that, but I do know that there are studies in CalARPA where they use the electron microscope to
I
try
know.
and see. So that was
I
also
know.
very cool to
I
find
know.
out.
I know, yes, well, we have one electron microscope in our labs, yeah. We
Amazing.
are not using it because it's so difficult to use.
Okay.
So sometimes you are right,
Yeah.
you need the training and you need. Okay, why do you remove the pigments?
At the time, we didn't have the confocal, so I had to make do with specifically let's focus on the light sheet, which I ended up using, and we weren't able to see past. I did add a picture. So this is the image that I took without. This was actually the first failed attempt at clearing, so it didn't clear properly. And this is basically just the surface level. You don't really see much deeper into the sample, so you don't see any of the structure, unfortunately. Yeah. Sadly.
No, but because what you were mentioning that if you are able to remove that part of the autofluorescence or whatever, maybe you can save that instead.
Yes.
I know that is nearly impossible to,
Yes.
but it could be another way to research as well in that sense, not to touch the sample or to touch the sample the minimum possible just to, to generate other artifacts that could be around.
I would, if I do continue this, I would also like to try and find different levels of clearing that protects the fluorescence. So you can see if you remove the fluorescence completely, what's the difference to that being towards clearing methods that don't remove the fluorescence at all. So that would be pretty interesting to see as well.
The last one, if I can. I think in the objectives you have something like to find the better stain combination. And the question is, what for?
Not necessarily a better stain combination, just more variety to see if there's any difference. If we use other stains, if there's more that we can see.
But in any case, what for? Because for me, a stain as an objective as well.
Yes. So for now, we were focusing on nuclei and filaments, but we don't really know exactly where everything is and how they are situated within the CalARPA. and so using different stains that focuses on different things, we're hoping to find more information based off of that. So that's the only reason why I want to expand the different stains. Yeah.
Okay, thank you.
Thank you.
Okay, thank you, that is very nice. So I just have one quick question. Let's say you have a new algae that no one has ever done any images from. Would you use these methods? Would you go right to the confocal? What would be the criteria for
Oh.
something not studied yet?
I think it would depend on what your level of expertise is. If you have background in confocal, go for the confocal. But if you don't have it, it's a lot easier to go to imaging platforms that the learning curve isn't that high. You don't need as many training hours to use it. And then, yes, I would do the clearing just because you can't see anything past the surface without the clearing. So if you have the confocal and you know how to use it, or you have the money to learn how to use it, then yes. But because we've also not really used it that much, we have a couple of images of Calata under the confocal. We also don't entirely know what we will be able to see. So maybe we see the structure, but we don't know if we're going to see information about the nuclei that much. So we could see the filaments for sure, but about the organelle distribution might not show up there. And then I would try and look for a way to. We actually did try to do live imaging when we do a cut on it to see what the flow looks like, but we were only able to do it on the Motic, not on any of the other ones, just because the second you try to zoom in, because this one is expensive, you can't do either of them without a cover slip. So you have the bottom cover, the bottom, I can't think of the word, sample that it has the convex hole, yes. So you thank you. You can put it in there and then put a top cover slide onto that. But then neither of these two can get past the initial stages. And so if it's a really deep sample, then again you lose a lot of the information, so you don't really see. And so we had some issues with that as well. We were very hopeful in the beginning, and then we tried it, and sadly didn't work.
Thank you.
Thank you so much.
Thank you.
Thank you.
The session, we move from the break. Congratulations.
Yeah.
We are live. We are live. Yes.
We are live. Thank you, Gab.
No chairs for you, okay? You're gonna be standing. All right. Good afternoon, everyone. We'll start the session of today. I will present you the jury. Well, first I'm Ana Maria Montegato from Ghent University as the chair of this session, and we have in the jury, Luana Montero. Wait, not yet, but the other.
The only one.
Exactly. And Lorenz Mechanet. We will start with Menahi, as he will present the study for the Belgian part of the North Sea metagenomic approach. You can start. I just want to mention for everyone in this session, you have 15 minutes for the presentation and the questions. You already know. I will stand up in the last 3 minutes of your talk just to make sure you know that you have to wrap it up with the conclusions. All right? We can hear you. The North Sea floor looks empty, but beneath every square meter of this sediment lies microbial communities that have been quietly running the chemistry of this system for millennia. And yet, we almost know nothing about them. My name is Nahil Fatma, and my master thesis is exactly about that. Microbial communities of the Belgian part of the North Sea, what they are doing, and what shapes them. So, marine sediments cover around 70% of the Earth's surface, and they are harboring microbial communities which are driving oxidation-reduction reactions and linking carbon, nitrogen, and sulfur cycle into interconnected system. And in doing so, they are regulating some of the key climate-relevant gases like methane and carbon dioxide. As you can see in this first block, we have mentioned like 3 cycles: nitrogen, carbon, and sulfur. And I have mentioned, of course, very simplified few regulatory processes across all the cycles, which are going to be the major focus of this study as well. But in order to understand how microbial communities are regulating these cycles, it is first crucial to know what shapes these microbial communities and what could impact them in the sediments. And it narrows down to 2 parts: environmental gradients and anthropogenic pressure. These were the 2 reasons compelled me to choose Belgian part of the North Sea for my study, because despite being very small area, Belgian Sea harbors different sediment types ranging from coarse sandy to muddy, and also it is one of the most heavily trawled areas in the North Sea, which makes it even more important to understand the impact of anthropogenic pressure on these communities. So, as much as we know about the macrofaunal communities and benthic biogeochemistry of this system, we don't know anything about microbial community composition in the sediments and their functional gene profiles for elemental cycles and which factors are shaping these communities in the sediments. So to address this gap, I chose shortened metagenomics because it captures taxonomy and functional diversity, and the good part, we don't even need to cultivate any microbes. This study spans around 3 research questions. The first, which is going to be answered in 2 parts. The first one is to understand the primary driver of community structures, either its sediment type or seasonality. And the second part is how that primary driver is going to be influencing microbial communities in terms of their taxonomy and functional diversity. Second is identification of dominant microbial taxa associated with key biogeochemical cycling processes. And the third, which is heart of this study as well, investigating the existence of a core microbiome across Belgian part of the North Sea. To answer these questions, a bi-monthly sampling campaign was conducted across 4 different stations in Belgian North Sea, and sediment cores were collected. And as you can see, near shore stations 130 and 700, they are muddy, and 780, which is offshore, is fine sandy, while 330 is coarse sandy station.
It is important for this study to keep one thing in mind, that station identity, which is 13700, is going to be used as proxy for sediment type, not location. And also sampling months is going to be used as proxy for seasonality. So in the lab, DNA was extracted from sediment corers and it was sequenced on Illumina NovaSeq platform using paired end sequencing, and raw reads were then processed through standard metagenomic squeeze meta pipeline, and at the end we retrieved 72 high quality maps, metagenomic assembled genomes, and downstream statistical analysis was being done in R. We can already get into the first question to understand the primary driver and its impact on microbial communities, but before getting into community level analysis, I first assessed sequence diversity by using non-parallel software, which is a reference-free assembly independent approach, which works on the principle of raw read redundancy to estimate community complexity across all stations. It generates curves for all the samples for every station, and it works on the principle the more complex a community is in terms of its sequencing, the more sequencing effort you need to achieve a certain level of coverage. And the more rightward will be the curve for that particular sample, and it's very clear. You can see that station 330 in the orange, it has the most rightward curve, and remember station 330 is the core sandy, which gives us the first hint that 330 core sandy type is the most heterogeneous station in terms of its microbial communities. What it also generates is a non-parallel diversity index, NDE, which quantifies community complexity as one single value, and that you can also see is highest for station 330. So this gives us our first hint that even at the broadest level, sediment habitat is shaping microbial community complexity. So the first part of the research question, to understand the primary driver, I ran PCOA and Permanova on functional gene profiles for all three biogeochemical cycles. Starting from nitrogen cycling, you can see the functional gene profiles were clustered according to stations, not month, and Permanova validated the result that stations explain most of the variables, not seasons. So the results were consistent throughout sulfur and carbon cycle. So here we established the first finding that sediment type is the main driver for structuring functional gene profiles. Season did not have any significant impact. Building on to this, why we are seeing sediment as main driver? Because sediment grain size controls oxygen penetration, depth, organic matter retention, and pore water dynamics, and these three things dictates redox environments, and short-term seasonal variations cannot override redox environment. That's why redox dictates the microbial communities. And how sediment type affect the structuring of community in terms of taxonomy? We have a community level bar plot. One of the most consistent result of this finding, Pseudomonodota being most abundant across all sediment types, across all stations, because it is highly versatile in metabolism. And also a huge attribution was from unclassified bacteria as well. But if we go a bit deeper into this graph, 8 out of these 15 most abundant phyla showed tightly sediment grain coupled trend. And I have plotted 2 in the line graph to show you the trend, which is according to the sediment gradient. Nitrosospirota are aerobic ammonia oxidizers, and they showed highest signatures at core sandy station. Here you can see. While thermodesulfobacteriota, they are canonical sulfate reducers, and they showed highest signatures at station 130, which is muddy. And we will see the impact of these results later. Now we know who is present here, we can get to the point at which biogeochemical processes are abundant across all stations. Starting from nitrogen cycle, the first step is nitrogen fixation, which is an anaerobic process because nitrogenase enzyme is highly oxygen sensitive. So it showed highest signatures at muddy station, while the next aerobic process, nitrification, which is conversion of ammonia into nitrate, showed highest signature at core sandy station. But one of the most counterintuitive findings of this study was denitrification, which is an anaerobic or anoxic process. It also showed highest signatures at core sandy station, which gives us the signal of copper nitrification, denitrification reaction going on at station 330. Next up we have sulfur metabolism. When organisms run out of oxygen and nitrate as electron acceptor, they shift to sulfate to harbor energy for their biomass buildup. And sulfur metabolism starts from sulfate reduction, which is an anoxic process, and it showed highest signatures at muddy station 700. While another important and interesting finding was sulfide oxidation, which is an oxic process. It also showed high signatures at station 700. So it gives us the hint for tryptic sulfur cycle, which is going on at station 700. Getting on to the last, carbon metabolism. Carbon fixation is one of the most constitutive and fundamental process that all organisms are doing to build their biomass, and you can see this process was not significantly different across stations because it's basic, while aerobic respiration, which is an oxic process, it showed highest signatures at station core sandy. Why did we see this particular overarching pattern across all biogeochemical cycles? Because muddy sediments at 130 and 700, they have diffusion limited water flow, which is a very slow process. They have consistent anoxic environment because of very high buildup of organic matter, which makes muddy sediments suitable for anaerobic phyla and reductive processes, while sandy sediments have poor water adductive flow, which does not let organic matter to retain at the stations, and they have continuous flushing of oxygen, which is making them suitable for aerobic phyla and oxidative processes to be abundant. So now we know who is present across all stations and which biogeochemical processes are going. We can get into the second question to know if dominant phyla across all stations are doing dominant processes across all stations. So this graph actually shows how much genes each phyla is contributing to all these processes, and as we have seen, Pseudomonodonta being more abundant in the blue in phylum bar plot previously, it is contributing most of the genes to almost all stations. But here I would like to mention some station and process specific trends, and you can see here, as we already saw in the process bar plot, in phylum bar plot, that Nitrosus phyota being aerobic ammonia oxidizers showed highest signature at station 330, and nitrification in the process box plot also showed nitrification was highest at station 330, and here you can see correlation that in purple Nitrosus phyota is contributing most to nitrification at station 330. So it gives us indication that yes, most abundant phyla are carrying most abundant processes across all stations. Another important finding is thermodesulfobacteriota, which are canonical sulfate reducers, but if you see in orange, they were highest at muddy station, but interestingly, they are contributing to nitrogen fixation at station 130 and 700. Normally, they are sulfate reducers, but they are contributing more to nitrogen fixation and also to sulfate reduction to some extent, which gives us an important finding that same phyla, same station, dual responsibilities, which shows these all processes and cycles are connected at one or another point. So this graph already tells us that the story that we have seen in the process level box plots and in the phylum level bar plots, they are same. Taxonomy is defining the function potential, and both of these factors are defined by sediment type. Moving on to the last research question, which is very important because this is the first time it was being identified, that if there is a core microbiome existing across Belgian Sea or not. So yes, out of 72 high quality mags, 26 mags were common across all sediment types regardless of strong sediment tide filtration in the different stations. Why is it important to know that there is a core microbiome? It shows, first is the biological reason that despite of all the changing and different redox condition across different stations, there is a set of microbes which is resilient to any change, and they are defining a functional backbone in Belgian part of the North Sea, which is going to be the basic biogeochemistry in the Belgian sea sediments. While another applied reason is, whenever we have to see the impact of anthropogenic pressure across a system, we always lean towards the core microbiome, because for example, if we have to see the impact of trawling, it's important to understand the working, presence and absence of microbes which are part of core microbiome. So that is why it's very important to have this foundation and to understand the organisms and their major functions. But as with all the scientific studies, this study also had few limitations. First we have conclusion. Okay, so concluding the study, sediment type, not seasonality, drives microbial community structure, and yes, we have seen sediment type shapes both taxonomy and functional potential, and we have seen an overarching pattern across all three biogeochemical cycles. And for dominant cycles, yes, dominant cycling processes were carried by dominant phyla across all stations, and both of these factors were defined by sediment type. And third, there is a core microbiome which is defining a shared functional backbone across all Belgian sea sediments. Moving on to a few important limitations. First of all, I did not have an even sampling design due to field constraints. Not all stations could be sampled across all months. And the other important limitation was not having enough and true replicates for all stations in all seasons. That reduced the statistical power a lot. But the good thing was large effect sizes compensated the reduction in power, because it showed that all the tests and all the biological differences that we have seen across stations in phyla and functions, they are still valid and robust. But in future, it would be great to have more replicates to increase the robustness of this study. Thank you.
Thank you, Vanessa. Very nice presentation, and you stayed perfectly on time. Thank you. We can start with questions with the jury. So let's go.
Okay. Well, thank you for your presentation. It's not really my area of expertise, so I will ask you some questions that might be basic to you. But so the first question, because it was a bit fast for me, the material and method. So I wanted to ask you, how did you process the sediment? Did you for all the sites you use the same sediment as slice dimension, or how did you do that? Can you recall me?
Yes, so basically we collected like sediment cores, and we used the core with the same diameter. It was 19 centimeter of diameter, and we collected top 10 centimeter layer of the sediment cores.
Okay.
But because actually this project was collaborated between three different domains, so for microbial study, I took the first 2 centimeter layer from that 10 centimeter. So the volume was same, so that 2 centimeter was used to extract DNA.
Okay. So you use this 2 centimeters.
Yes.
And okay. Did you have some idea of the let's say how was the oxygen profile in all the sediments?
No.
Oh, okay.
Because I was not part of sampling campaign, and we could not get the real-time measurements for oxygen and organic carbon content, so that was a limitation for me, that I did not have oxygen or vertical profiles for sediments.
Okay. But yes, but you know that there is this in some sediments, it depends on the bioturbation of the site, but you might have some structure with the different processes that are at different depth in the sediment.
Yes.
So depending on the oxygen penetration depth, you can have a site where you have all the processes onto, I don't know, fermentation, but at other sites, maybe in 2 centimeters, you stop at the nitrogen, reduction depth. So my question is, how can you use that information when you compare the microbiomes at the different sites? Is there a trend that goes along with the observation of the genes that are present that you could infer for that too?
So normally, Belgian part of the North Sea is not very deep, but the thing is, the first thing is for sure we had like equal depth for all the sediment types, like 2 centimeter, but these are not all the processes that I had. It was like a lot in the manuscript, but I had to shortlist the important ones. But the trend is same because 130 and 700, these are muddy, highly muddy stations, and they are anoxic. Even if you see their color, they are dark in color. So mostly reductive processes which are anoxic are high there, and it is also very well documented in the research. But this 780 is fine sandy, and pore water reductive flow is better there, and the 330 is even more coarser, so it has like more spaces between the sediments. And based on those profiles, it was like very clear trend that productive processes were abundant at 130 and 700, while oxidative processes which needs oxygen, for example nitrification, they were abundant at coarse sandy stations. And they were like correlated with the gene presence across stations and the processes.
Yes. Okay. Okay. So now I will ask you another question. If I understood, you are identifying genes, and when you compare the occurrence of the genes, like maybe you can explain that to me a bit better. You have the number of markers for specific the genes that are marker for specific process. The number of markers, they are the different kind of genes. Yes. How can you relate that to the intensity of a process? Like, how does that? How can you transform that into the intensity of the process that after that, let's say, oxidize the carbon, for example,
I
the
get
carbon
your point.
flow or whatever.
But the thing is, with this research design, we cannot quantify the process because for that we need biogeochemical flux measurements. That was one of my questions initially, but we did not get flux measurements due to technicalities. So this research design based on only DNA extracted, we can only understand the presence and absence of a process and gene across stations, not the intensity. For that, either we have to go for metatranscriptomics using RNA, or we have to use real-time biogeochemical flux measurements, which I did not have. So it's just presence and absence, a trend.
So it's like the potential.
Genetic potential, yes.
Okay.
Thank you, Lorenz. I think we need to pass to the next to remember because of time limitations.
Yes, thank you for your presentation. I want to talk about the sampling design. So you have 4 sessions. Obviously, time is limited. You cannot do everything. But you say that you have 4 stations that differ according to the sediment type.
Yes.
Which makes perfect sense. But they probably differ by many other aspects as well. Did you consider that? And what is the yeah? Can you have a critical view on this choice that you've made?
Yes. First of all, the most critical point that we realized quite later in the research was that because I did not have replicate, so it's really not possible to separate the impact of location with the sediment type. Although I do have several lines of evidences in my research that show that sediment type is overarching than the location impact. But yes, the very first thing is it's very important to have replicates in each station for each month, because technically, for example, for 130 I had 1 sample in April, 1 sample in June, but technically I should have 3 samples in April and 130, 3 samples in June, but that was not the issue, that was not the case. So that is the most critical point, not having replicates, but I can still show some evidences that are very clear showing the sediment type is the major.
Yeah, I completely agree. I mean,
Yeah.
you're probably right, but I mean, you could have given the same presentation, saying I have 4 stations that differ by their distance to shore or by their depth. Of course, sediment type makes more sense, scientifically speaking, but technically, there's no reason to choose that. Or and just another question: can you go back to the carbon cycle? I was a bit surprised by this cycle. Yeah, what is carbon fixation?
Carbon fixation is basically when carbon dioxide is being converted into organic carbon.
Okay, who does that?
Uh,
I mean, that's a reaction I'm quite familiar with.
yes,
And I call it photosynthesis.
normally all microbes fix carbon, like they can, because this is mostly like dark carbon fixation, not like in the light part.
Chemosynthetic?
Yes. So almost all carbons does that, or microbes do that because it's a constitutive process. That's why I did not get any significant difference across stations.
All microbes don't have this ability to, I mean, there's lots of heterotrophic microbes. Yeah. Okay, thank you very much.
Just checking.
Yes, you can
My
say.
my
And
time.
we can hit you in.
When I hear congratulations, really excellent presentation. You got a lot of important information from your project. I'm acquainted with a lot of challenges that it is to do this study. But yeah, environment, many variables, many things in place. We'll try to put two questions for consideration to hear your thoughts on it in one. And do you think it would be a feasible goal to, when, if you can expand, and what would you do if you can expand the study, if you would have all the resources, not many limitations on choosing, expanding more stations, more sampling, all resources, people do it, doing all the work for the gene extractions and a lot of time to do that. And what is more important, what would you like to do? And if you could achieve to use the picture of the microbiome diversity and composition as a proxy for other benthic fauna.
Okay, so for the first question, what I would like to do if I had all the resources is I would like to have more replicates, and also I will not just stick to the DNA-based presence study, but I would like to incorporate metatranscriptomics or at least biogeochemical fluxes, because that is real-time calculation. And also one thing that I would like to incorporate is the impact of bioturbation, macrofauna, because macrofauna do impact a lot the working of microbial communities. But due to data limitations, I did not get data for macrofauna bioturbation activities. But I would like to incorporate as a separate layer in this work to understand their impact on microbial communities, and then how microbial communities in turn are reacting to all these reactions or biogeochemical processes.
Do you think there is other benthic compartments and macrophagas that have a strong relationship with the bacteria that could that are relevant for understanding all the biogeochemical
Like what are those?
chemistry of the sediment?
Yes, there are macrobenthos and all the like worms which are doing reworking in the sediments. That is controlling oxygen penetration, water penetration depth, and also organic matter retention. So I don't have much knowledge about macrobenthos because I didn't focus on that niche. But yes, I'm sure because I have read in literature that macrobenthos do impact a lot sediment habitat redox conditions, and that we have seen is the main interpreting factor for microbial communities.
Thank you.
Thank you. And Olivia?
Thank you, Anaël, for the presentation and the nice work. Could you go back to the PC one on functional? That's the starting point, Gauthier, don't laugh. It's not a statistical question. The first one you showed.
For nitrogen only?
Yeah, okay, so what are your variables here? behind this?
station and month.
That's not variables. But what is a PCOA? that look-
Functional gene profiles.
Okay, so okay, because we're going very fast. So you have all your, and do we know anywhere in each of your functional type, like nitrogen, cycle, etc., how many of these
It
functional
is
genes you
my
have?
manuscript, like a long list of marker genes that I had to extract from the bioinformatics part.
So there's,
There are plenty of marker genes for one.
yeah, I understand, but just order of magnitude, is it like 15 or is it 1200?
No. Like maximum for one process, there were like 5 to 6 genes,
Okay,
and they are TPM values that I took from the bioinformatics, and then I used them as.
okay, because when we look at, I see that it's there's like 85, 90% in 2D. There's not so many variables, but we don't know that at this point. And related to that, like so here we see, I guess you use Breakertis or it's Breakertis, but the data is presence absence.
Data interpretation is presence absence, but data itself is not presence absence because it is DNA based study. DNA only tells us the potential, if potential for
Yeah,
this
but-
process.
So in the table there is actual values, not just ones and zeros.
No.
Okay. So okay.
There are like TPM values, transcript per million. Like you can say those are percentages, relative percentages of genes.
Okay. And what does that bring us different from the stacked bar plots or the box plots that you present later? Use it on the same, because it's the total number of genes at every time. Here we have the actual genes,
Yes.
but it's difficult for me to see really what's going on in between the two, and also you're using absolute values.
No, relative.
Where?
There is no absolute concept. It's always like relative percentage because we always have an
Well,
issue.
if you go to the box plots, that's relative values,
Yes, these are like relative abundances.
but relative to what? You're gonna stop me, okay? Just ask, but
For
relative to
example,
what?
if there is one sample and I know there are like 2 genes,
Yeah.
gene A and gene B, and if I see like gene A is 50% or gene B is 60%, and if I compare it between 2 samples at same station,
Yeah, but-
I really don't know if gene A in sample 1 is increasing or gene B is decreasing.
But you have some of the other graphs with the box plots, it goes way over 100.
Yeah, but these
So
are,
that's not relative.
this is, so technically, it's like some of the TPM values. Here I summed up the TPM values for each process at each sample, for every station.
No, I think that what makes it difficult is that I think they're absolute values because it's over 100. And what I would like to know to better understand the importance, even if it's just a potential, as you said, of each process in each station, I would need to have a relative, or I need to have I cannot compare these values because the 150 for electrification in the coarse sandy might be like 10%. But it looks like it's the most important there, and maybe the 40 in the muddy is like 90%, but we don't know, and that's really difficult for
This
me
is like
to understand.
that I had to address compositionality of data that we could not do. I did the lab part for it by using external genomes, which we call it standard internal genomes.
Okay.
We add known quantity of external genome from a bacteria to know how much genes or how much DNA is coming from each bacteria in the sample for a particular process. That was an issue of compositionality of data here, but I could not process those samples for bioinformatics.
Alright.
But you are right, you can never know if it is one process is decreasing or the other is increasing. That is relative issue, and it's always there in ecological microbial studies. But that is the next objective for this project.
Thank you very much.
Thank you for bringing up that point because it is one of the most important issues.
Thank you.
Okay. We move on to the next one, which is Carolina. She's going to explain us about nudibranch and how we're going to Let's see. Well, I let the floor is yours.
Yeah, before starting, I wanted to show you that I think that my thesis it's Italian culture. So I would be happy if you wanna see it during the presentation, discover a bit more, and go better into details. So first of all, thank everybody for being here. Good afternoon. I know it's quite late, but you're a lot, so thank you. I'm Carolina Argnani, and for my thesis, I investigated environmental heterogeneity as a nudibranch driver, as a driver of nudibranch assemblages and their chemical profiles. I did this thesis during the 5 months I spent at Unirio in Rio de Janeiro, Brazil. First of all, let's define what nudibranchs are. Beyond their appearance as fantasy-like creatures, nudibranchs are mollusc gastropods, which names come from the Latin nudus, which means naked, and the Greek branchia, which means gills. This is because of the absence of a protective shell and the presence of gills as their primary respiratory structures. Nudibranchs inhabit a wide range of benthic marine environments, from shallow coral reef to deep sea habitats. And to compensate for the absence of a protective shell, they have evolved chemical strategies, meaning they use secondary metabolites that are either sequestered from their diet or created de novo to render themselves toxic and unpalatable to potential predators. Nudibranchs also display xenophagy, meaning they have a hyperspecificity and low dietary plasticity. Consequently, diet and food availability act as a driver of both spatial and chemical ecology. So this prey specificity and their vast taxonomic diversity mirrors in a broad spectrum of associated preys, including sponges, cnidarians, rhizomes, and tunicates. So nudibranchs exert a top-down regulation on these benthic communities, acting as a biological control and maintaining the ecological equilibrium of these benthic populations. So this also has a conservation relevance, as a decline in a specific prey population could lead to the localized disappearance of the associated nudibranch predator, and consequently environmental characteristics and alteration that affects food availability, like temperature, pollution, and substrate composition, also affects nudibranch spatial and chemical ecology. These characteristics, paired with their short lifespan, underscore the potentiality of this organism of being bioindicators of potential ecosystem health, enhancing the importance in investigating their ecology. So in this study, field surveys, computational analysis, and chemical characterization were involved to address 2 research questions. How does environmental heterogeneity shape nudibranch assemblages and their chemical profiles across different taxonomic levels, from inter to intraspecific variation? The environmental heterogeneity is conceptualized across 3 hierarchical scales: a macro scale that considers environmental variables like temperature, pollution, and salinity and depth; a mesoscale including substrate composition of the sampling quadrant; and a microscale that considers the rocks within the sampling quadrants, and in particular their area and their coverages. Although the recognized importance of these environmental variables the studies examining their combined effect on nudibranch assemblages and on their functional traits, such as secondary metabolite profiles, are still limited, especially in the southwestern Atlantic and in Brazil. So this research aims to address this knowledge gap. The study took place in Arraial do Cabo in the state of Rio de Janeiro, and in particular in 2 beaches, Prainha, characterized by low anthropogenic impact, and Praia dos Anjos, which is subjected to higher anthropogenic pressure due to its proximity to a local harbor. The area of Arraial do Cabo experiences a coastal upwelling phenomenon typically occurring in Praia Grande, in which South Atlantic center water brings nutrient-enriched and colder waters to the surface, enhancing the productivity and the biodiversity of the entire area. For each beach, 15 quadrants of 1 per 1 meter were placed, and its coverage, its substrate coverage was being recorded with a camera. Then each rock within the sampling quadrant bigger than 50 centimeters was picked
turned and investigated for nudibranch presence. Then the underside coverage was also recorded. When finding nudibranchs, these were placed in jars and kept separated between each other to avoid cannibalism. Then the jars with the nudibranchs were brought to the base laboratory where each specimen was put in an aquarium and be recorded individually to allow for morphological identification. Also, the length measurement was performed, and one representative individual per species was preserved in alcohol and donated to the National Museum of Rio de Janeiro, while the subsequent individuals were stored in freezer for the chemical analysis. Computational analysis was conducted using the software CoralNet to assess the percentage coverage of the substrate, so the quadrats, and the underside rocks, while the rock area was calculated using ImageJ. Statistical analysis was performed to address the first research question, if you forgot, about environmental heterogeneity influencing nudibranch assemblages. To do so, community-level parameters were considered, and in particular, abundance and species richness. Firstly, the predictor variables showing collinearity were removed by testing them using Pearson and linear regression, and generalized linear models with Poisson distribution were performed for both abundance and species richness, followed by a model selection by a kite information criterion adjusted for small sample sizes to identify for the adequate set of models. A qualitative comparative chemical analysis was performed to address the second research question, so about environmental heterogeneity shaping chemical profile variation. And to do so, a customized protocol was developed by the team. The chemical analysis was performed at UFI, Universidade Federal do Fluminense, in Niterói. And each specimen was, the chemical compounds of each specimen was extracted independently to allow for individual level comparison of the chemical profiles. The chemical profiles were resolved running 3 thin layer chromatographic plates using different ratios of hexane and ethyl acetates as mobile phases, establishing a polarity gradient. Cholesterol standard was used as an internal control for all the 3 plates. And firstly, the plates were inspected under UV light and subsequently treated with serum sulfate reagent and heated to show more chemical bands. So let's go to our results. Prainha displayed a higher mean sea surface temperature and a greater variation of each quadrat compared to Anjos Beach. A similar trend was observed for salinity, while the bathymetric distribution of each quadrat resulted more heterogeneous in Anjos Beach compared to Prainha. Regarding the pollution, the fecal coliform level were considered, and they resulted being below the threshold for both beaches, indicating good water quality. So it was not included in the subsequent analysis. Then the substrate coverage resulted in Prainha being with a higher contribution of biotic structure, with algae being the dominant coverage, while Anjos Beach had a more abiotic contribution, although algae coverage, which is this green slice, was still abundant. At the underside rock level, Prainha displayed a higher dominance of yellow encrusted sponge, represented by these pink slices, and Anjos didn't show any clear dominance of coverages. In total, 25 individuals of nudibranchs were collected, belonging to 9 different species in both beaches. In particular, 9 individuals and 4 species were collected in Prainha, while in Anjos, 16 individuals and 8 species were found. 3 species were collected from both beaches, this 3-1, while only 1 species was just found in Prainha, and the other 5 species were collected only from Anjos and not in Prainha. So the statistical analysis, the generalized linear model for Poisson distribution for nudibranch, with Poisson distribution for nudibranch abundance showed no overdispersion and confirmed an adequate fit to the data. In particular, the top-ranked model included water temperature and rock area range as the best predictor for nudibranch abundance with statistical analysis. In particular, temperature showed a negative coefficient, while rock era rings show the positive one related to nudibranch abundance. For the chemical analysis, the ecological characterization of the potential food source coverages on those rocks exhibiting the nudibranchs that were used for the chemical analysis showed that some rocks had a biogenic heterogeneity in food source composition. Some others had a homogeneous coverage, as we can see this is just yellow encrusting sponge. And few rocks display a total absence of potential food source, but still nudibranch presence. These are the thin layer chromatographic plates. This color one are the one treated with serum sulfates reagent, while this one is the UV inspection. Each number represents an individual. And in general, from the first plate, in the upper portion, which is the more apolar one, we can kind of see yellow bands that are shared across different species and are characteristic of fatty acids. Another band that was broadly recognized was this one, circled by the purple circles, that was also shared across different species and at different intensities. And it was at the same polarity range and at the same color, with the same color as the cholesterol, indicating a sterile nature. Then regarding the UV inspection of the first plate, we saw bands that were not visible in the respective plates treated with serum sulfate, these red lines, indicating the presence of unsaturated molecules. From the second and the third bands, more bands distinct from the chemical profiles, enhancing both the inter and intraspecific difference of the chemical profiles, and especially the difference in chemical profiles of individuals belonging to the same species, and also found on the same rocks. So there were different chemical profiles. So what can we say about our results? That this study we believe this study brought a significant contribution to both global and local repositories, and it provided local and precedent records of some species in the area of Arrayal do Cabo. Then if we look at the nudibranch density, so individuals per meter squared of sampled area, and we compare our studies with the others worldwide in hotspot of biodiversity, we can see that these this study brought a rigorous sampling methodology that could overcome the visual biases represented by the previous studies, the methodologies of the previous studies, for example, linear transects, and it could contribute in diminishing the underestimation of benthic macroinvertebrate communities. Regarding the negative correlation between water temperature and nudibranch abundance, this can be correlated to the thermal tolerance of these animals. In fact, higher temperatures induce a metabolic stress to the nudibranch, impacting on their growth, on their reproduction, on their feeding and reproduction. Consequently, low water temperature supports nudibranch abundance and reproduction. The positive correlation between rock area variance, so different sizes within the quadrats, and the nudibranch abundance emphasize the importance of architectural and structural heterogeneity in driving nudibranch spatial distribution, as a higher heterogeneity provides more spatial niche and microhabitats, also providing more surface for feeding, for deposition, and also for refugia from predators, and it also contributes in minimizing the localized competition. This positive correlation paired with the negative correlation between nudibranch assemblages and coverages like substrate coverages or rock coverages can be correlated to the mobility patterns of the nudibranch. In fact, in this study, we presented a new untested hypothesis of high mobility of these nudibranchs, for which in highly diverse habitats, these nudibranchs can move more and allowing to encounter more food and diminish the predation and the competition. So although a greater sampling effort is needed and further chemical analyses are needed to investigate the environmental heterogeneity and the chemical compounds that were not detected, we still found inter- and intraspecific chemical variation, although their possible wider home range might decouple the correlation between their chemical profiles and the specific microhabitat, so in this case the rocks, that they were located on. Conclusion and take-home messages. We found different assemblages in the two beaches. We saw that multi-scale environmental heterogeneity influenced nudibranch assemblages and chemical profiles, potentially chemical profiles. We presented a new hypothesis of high mobility of animals. Oh, sorry. And this study represents the first study on nudibranch assemblages in Arrayal do Cabo. And it also provides rigorous and standardized protocol for both sampling and chemical extraction methodologies, which can be used worldwide and also for other macroinvertebrate species and nudibranch population. Assemblages studies. And thanks for the attention.
We'll start the other way around this time.
With me?
Yes, careful.
What do you mean?
No.
Not a worry. Thank you, Karai, for the presentation. Interesting study. I like the fact that you start by planting at the multi-scale that you're going to work at the site level, etc. Can you go back to I have a question about the first result you showed. I think it was the temperature.
Yeah.
And you don't show us the rest, but you show us that. And I'm wondering if the rest is similar. Is it like the site that's important, or do you have like in terms of temperature? I would say we have two different habitats.
Yes, thank you for the
in
question.
the two sites.
Because I wanted to point out as well. I'm gonna go back on the sampling part because it is important, the sites. As I said, the quelling phenomenon typically occurs in Praia Grande, and we can see that Praia Grande is closer to Anjos than in Prainha. So we think that this lower temperature that was in Anjos can be related to the position that typically occurs, not always, but typically occurs for the upwelling phenomenon from the Antarctic waters.
Okay. And did you have the same divide for the other variables that you say it's the same general pattern? So I guess that's the median.
Salinity had a similar trend, but it was not significant in the statistical analysis.
Okay.
So that's why I decided to put just temperature, and the bathymetric distribution had similar patterns, but the opposite. But it's because of the sampling we did.
But the sites that were like the few very cold sites from Quadrat from Prainha are not also different than the other variables. So you didn't see a pattern there.
No, we didn't.
Okay, so it makes sense like that. You said the technique you use in this study maybe allows you to solve the problem of maybe under sampling in other methods, or was there a way to has anyone actually compared them on the same sites at the same time? Do you think it'd be possible to actually confirm that?
I think it would be possible. I will again go over and over.
Yeah. Perfect.
This one, because for example, in these two cases, they did this study in the same place, in the same sites, and in different years. So this is more recent, but in this case they found less thing, but they use the same methodology.
Yeah, but the sampling surface is 4 times larger, and maybe they sampled non-habitat for that species,
But this is high biodiversity, it's an hotspot of biodiversity. I believe that also the methodology can be involved in this, because they just did linear transects and they didn't even, if I remember well, they didn't even turn the rocks. And also, this is another thing that is interesting, because not always nudibranchs are below the rocks. Sometimes nudibranchs of the same species in another part of the planet are on the surface, of a reef. I remember when I dove in Portugal, there were there. I didn't have to turn any rock.
Okay.
And he said in Araya do Cabo, it was impossible to see them if
you're
not
doing
turning.
it. Okay. For now, I don't have any more questions.
Sorry?
I don't have any more questions for
Okay.
now. I told you 3 minutes.
Thank you, Anna. Carolina, congratulations.
Thank you.
Very interesting presentation. Extremely important and novelty record. So great contribution to science. Could you, well, two considerations, I will see if I manage to track my time to leave time with my colleagues. Can you show, go back again the map of the study site?
Yeah, of course. These ones.
Yes. Do you have any idea, apart of course from the upwelling, which is indeed extremely strong there, the position of the continental shelf. This current circulation of the surface that to try to explain the hypothesis that you mentioned before.
Okay,
And it would influence the temperature in Anjos?
You mean like the grade of the surface that can bring effectively the colder waters to Andes? I didn't check about it actually. It would be interesting. Yeah, but I guess it also changes by the season. But yeah, I didn't explore in that sense to see if actually the waters from Praia Grande were reaching more Andes than Prainha. Is this the question you were?
Yeah,
Okay.
because
But that's interesting, yeah.
that you mentioned, there could be the temperature in relation to that, or the climate in that the time that you took the data, could that have an influence in terms of if there was more rain or not in the topography of Anjos versus the Prainha?
It was the temperature and the climate was basically the same because it was done in 2 weeks, the sampling.
And in terms of rain?
There was a rain in the middle of the sampling campaign, yeah.
But do you think, because I don't remember by heart, different know the area, but could there be differences in the geography of the area that the rain input could have an influence on the observatory?
I don't think so.
Water temperature?
I don't think there will be a difference between the two beaches as they're at close.
If I may, one last thing. You mentioned Anjos being, if I understood correctly, suffering more anthropogenic pressure.
Yes,
compared
because there's a local harbor here.
Yeah, compared to Praia. But does that mean that Praia is kind of preserved, or is there also high pressure because of tourism, for instance?
So the pressure in Prainha is increasing because of tourism. I cannot tell you if it's more than Anjos. I think Anjos has way more anthropogenic pressure because there's a lot of boats that passes every time. And in Prainha it's more like tourism, and for what I saw, it's more physical damage on the beach than a pollution damage. So I would still think that Anjos is more in a pollution way impacted, although it's still the fecal coliform levels and concentration was below the threshold, so it was not like a worrying situation at the moment. At that moment, yeah.
Thank you.
Thank you. Thank you for the questions.
Thank you, Carolina. I have some questions about habitat heterogeneity. What do you exactly mean by habitat heterogeneity?
I mean the differences in the habitats. So I will go back. So for example, in each quadrant, we wanted to see if the temperature, the pollution, the salinity, and the depth regarding the macro scale was influencing the nudibranch assemblages. So if, for example, they preferred higher temperatures, or they prefer higher depths, or they prefer higher salinities. And also for the substrate composition, for example, we sampled the coverages, so algae coverages, coral coverages. So we wanted to see if each coverage, algae, coral, spongy, bare rock, was influencing again the nudibranch assemblages. And the micro scale, it was more at the microhabitat, the specific microhabitat they were located on, so the rocks.
Okay, but I'm not sure I really understand the difference between substrate composition and rock area.
I will show you. So this is a substrate. The quadrat coverage, and we sampled by we assess the percentage by recording like this, and then with the coral net we assess the percentages by making 120 points per image. And this instead is the rock. So we were turning the rock, and the coverage of the rock was the microhabitat, the micro scale heterogeneity. This was mesoscale and this was micro.
And perhaps another question about this mobility hypothesis that you came out with.
Yeah.
You say there's nothing to eat. Are you sure there's nothing to eat for them on the rocks?
So
why would they?
Because
Why
we
would they come to this rock. I'm not gonna make it to a restaurant that does not offer me food.
Yeah.
I mean, what we related was seeing on literature the possible food sources.
What is this orange thing?
It's oxidation of the, and this was also,
Iron oxide.
and this was a wood base. So we know that this species that was found likes wooden rocks, but because of the refugia. So because there are a lot of holes in the middle. And in fact, that's how we found them. We turned the rock, there was no one. And then from the small holes, they started to come out 3 nudibranchs. We found 3 nudibranchs of the same species here. So.
Because you see some sponges and hydrates, but I mean, I saw in your photos some Aplysia. Aplysia is a pure herbivore. So you can also expect Aplysia just to feed on the biofilm, and you wouldn't be able to see it, and yet it's good for them.
In this case, we didn't find Aplysia. We found Anax jubatus.
Thank you very much.
Well, thank you.
Yeah. Congratulations for this first study on new differential in the area. It's good that you did it.
Thank you.
And it's nice for us to see the picture also. I have it's more a comment on your again those 2 study sites. The part that is the northern bay.
I will go back so it's easier. You mean this one? The Peña.
Yes. Is it the one where the temperature is higher and the depth is lower?
Yeah, the depth of the sampling quadrants was lower, but not lower.
But is it also the fact that the depth is lower? Is it for all the bay or like?
No, it's just for how we sampled.
Okay.
And also for placing the quadrats, we had some criteria. So like not too much current to make the quadrats oscillate. And we also took care about the corals to not damage the corals too much. So enough rocks to pick. Yeah. Like if there was, it was like very deep, but no rocks, we didn't.
Because I was wondering, and the fact that there are tourisms going there is also telling me maybe this is that when you have a close bay with a small depth, then the radiation is hitting the water more, which is good for biology, of course. But maybe this is also what happens in this area to explain the temperature. It's more like a problem of the radiation of the sun that is not evacuated by the circulation. That's why maybe
I think a greater sampling effort would be needed,
Yeah.
with different depths to actually see, because also the alternate models showed that included depth as a predictor, but not with statistical significance, as a predictor of new difference abundance again. So there is a trend, but it's not statistical significant, so we still don't know. But it would be interesting to discover more and to see each variable, how it influences.
And then you discuss thermal stress.
Thermal, yeah.
Yes. And you have this temperature variability from side to side. How do you know what is the value where thermal stress is starting for the species that you observe in relation to the temperature that you have measured?
So I don't know, but from my memory, looking at when I did literature review, I didn't see that many experimental ecology studies. Also because these animals are really hard to breed and to do an experiment on it. In fact, in aquariums they are not used because they die instantly. As soon as you take them out of the water, after a few hours they die. So I think doing an experiment on thermal stress can be hard.
Okay, thank you very much.
Yeah, it's just
And last but not least, we have Diego, last presentation of today, so stay awake. He's gonna bring us to another exotic topic for Ecuador and the mangrove forest over there. The floor is yours. Just to warn you, because we hear the applause for next room, okay? Don't stress, because we are already some minutes in delay. So I will give you my sign when your timing is.
Awesome.
Yep.
Thank you. Dear members of the jury, dear friends, dear colleagues, I'm delighted to present you my thesis work today, entitled From Tidal Flood to Mangrove, a Biodiversity Assessment of Faunal Dynamics at Aquaforest in Ecuador. So today I'll be talking a lot about mangroves, and I want to give you a little bit of context about what is this ecosystem. So Mukerji et al. Defined mangroves as plants, often woody trees or shrubs, that grow along the intertidal land-sea interface, and together with their associated organisms and abiotic factors, they constitute the mangrove ecosystem. And what does this mangrove ecosystem do? They provide a lot of ecosystem services, such as the protection of coastal populations from erosion and extreme events. They store carbon, they help regulate the climate, they enhance the biodiversity, they have socioeconomic benefits, and so on. So the list of ecosystem services is really long, and that's why these mangroves are so precious for both us humans, but also for a wide variety of coastal organisms. Yet, these ecosystems are threatened worldwide, and 50% of them are at risk of collapse by 2024, according to IUCN. And these mangrove loss are mostly driven by anthropogenic impacts, as always, such as wood harvesting, aquaculture, and urban development. And more specifically, in my research area, shrimp farming was identified as the primary loss driver. And that's exactly where sits the project I worked on, which is called Aquaforest. It is an eco-engineering approach, and its concept lies on the circular reuse of dredge sediments that are placed where they were modeled to deposit naturally. I'll explain myself. So Reno and Al observed that in the Guayas River estuary, tidal flats were being formed due to anthropogenic causes. So Jan de Nul, the big engineering company I did my thesis for, saw an opportunity in this phenomenon and they decided to elevate a 50-hectare substrate by approximately 1.5 meters, and then after that to plant some mangrove rubagles and saplings. So basically they designed a completely new ecosystem. And I really like this statement from one of my supervisors, Professor Reyes. She said during an interview that Aquaforest is a living laboratory where you can really see how a new ecosystem develops and forms. And that's precisely why I feel like that Aquaforest is a unique opportunity to understand the fundamentals and the fauna dynamics of such a new and precious ecosystem. So to move into my hypothesis, the first one was that the biodiversity at Aquaforest will increase over the monitoring period. Secondly, that Aquaforest biodiversity develops positively relative to its pre-construction state, so when it was still a tidal flat. And third, that Aquaforest reaches a viable ecological state and converges toward the biodiversity of a natural mangroves in the estuary. And to do so, local experts monitored the pre-construction state of Aquaforest. They monitored reference mangroves, and of course, they monitored Aquaforest. So to dive into the study area, my research area is sitting on the Pacific coast of South America, more precisely in Ecuador. And as you can see here on the map, this is the Guayas region that is marked by really high seasonal variability. So we can really see a wet and a dry season that starts and stops really clearly. And this estuary is tide dominated. So we observe up to 2.5 meters of tidal range at the Aquaforest site, which is just here. So this is quite a lot. And more on the country level, Ecuador and this region is subject to ENSO extreme events. So that's El Nino and La Nina events. That's actually happening right now. So, yeah, it's impacting a lot the region. For the references, we have B1, B2, B3, and B4 that are reference mangrove that were used to compare data of Aquaforest. They all mature and develop mangroves, and they are within the estuary. More precisely to our Aquaforest site, it sits within a protected area, and 3 native species were chosen to be planted on the newly created substrates. And the first results were super impressive. There was a 90% of survival, so that was really encouraging for the rest of the research. To move into our data collection, there was a multi-taxa monitoring every month from February 2025 to March 2026, done by ESPO, this is the local university, and 3 stations were monitored. So we have P1, P2, and P3, and at each station, 3 methodologies were used. So first of all, we had the Van Ven grab, trying to characterize the subtidal community. Then we had quadrats, looking more at the intertidal community. And last but not least, the point count methodology to characterize the bird community on the substrate using binoculars. For our data analysis, every analysis were done using R and using the vegan package. And for all the samples, diversity indices were calculated, such as species richness, the channel index, evenness, and the average taxonomic distinctness. And more specifically for the microbenthos, we had a look at the beta diversity with the breaker-list summary index, and for the avifauna, which is the birds, we looked at the dissimilarity using the Jacquard, and that was also partitioned into turnover and nestedness. So for our results, first of all, we had a total count of 912 individuals representing 68 taxa. And if we have a look at the graph here, we see that P1 and P2, that's the community, the class composition. P1 and P2 are quite even. They're bivalve and polychaete dominated, whereas the third station, P3, is completely different and is gastropod dominated. For the subtidal community, unfortunately, we cannot say much. Unfortunately, the samples did not allow us to characterize really well the community. community. So we chose to not compare them to our reference mangroves. If we focus a little bit more on the intertidal microbentic community, the composition of this community differed among stations, but also between seasons. And again, if we look here at the LMDS graph, we see that P1 and P2 are quite similar, whereas P3 is different. And these differences are likely due to the fact that the stations on aquaforest are heterogeneous. We have a different sediment composition. We also have different elevation on the substrates, which leads to different inundation regimes. So that might have had impact on the community composition. For the bird community, we had an impressive total count of 8,000 individuals, representing 53 species. The taxonomic distinctness was quite high, highlighting the broad taxonomic spread of the community. And if we look at the family level, we see that aquaforest and the four references are dominated by herons, egrets, and ibises, which is typical for a mangrove community. Whereas the baseline study, so when aquaforest was still a tidal flat, it is more dominated by species which are typical from a tidal flat. Across my data analysis, I realized that aquaforest was the richest of all sites with 19 species, and I thought that was not really logical. So actually, I realized it was just that aquaforest is, like on the family level, is converging towards, it's trending towards a mangrove community, but with still carrying its tidal flat background. So we can say that the family and the guild structure is already trending to reference mangrove, but with its tidal flat background. If we look more in depth at the species level, we see that the reference mangrove cluster together, so which is a good sign of similarity, and that the baseline is completely different from the mangroves. And actually, aquaforest sits midway between these two types of ecosystem. Here's another look at the same data. If we focus on the third reference site, we see that aquaforest and the third reference sites share already 6 species, which is more than the 3 other references. So that means that the third reference site is the closest, the most similar to aquaforest. So at the species level, we can conclude that aquaforest is still distinct from the references, but is also distinct from the tidal flat. So it's definitely evolving. So for my discussion, first of all, the first target was met. By June 2025, the plantain stand was established with a 90% survival. That was a really good result, more than expected actually. And the group that had the clearest signal was clearly birds. We had a 43 species over 13 months, which is quite a lot, and that is due because of the characteristics of birds. They are highly mobile, they can colonize the substrates quite fast, and we even recorded some breeding activity on the site, so it's really encouraging for the project. For microbentic community, we saw that the intertidal richness doubled. It went from 3 individuals per quadrat to 7 individuals. And it takes more time than birds. Obviously, the recruitment is more steady than the birds. So yeah, in conclusion, the engineered substrate was established by both fauna and flora. For the trajectory towards reference mangrove, on the bird point of view, we saw that family and guilds have converged. Now aquaforests match partly the regional aquafauna mangrove community, but we cannot say as much at the species level. But this is normal, obviously. The dissimilarity from the references, for aquaforests with the references, is driven by turnover, whereas with the baseline is driven by nestedness. And for the macrobenthos, unfortunately, our data was too poor to draw conclusions, and I think it's also due to the fact that the physical setting on aquaforest is too variable to have any conclusion. So the trajectory is positive, but it's really too soon to call it a success. And to have more a look at the climate point of view, right now, especially right now that the El Nino started, aquaforest is at a very exposed stage. It doesn't have the root system to really bind the sediments as well as much as a nature mangrove can do. It builds in its own resilience usually, and aquaforest is still a young mangrove, so it sits at the very start of this feedback loop. There was some engineering that was done. They built a bamboo wall that they buried to help bind the sediments, but this was done to buffer from waves created by the boats going in the canal, and not to buffer some super El Nino river discharge and sand deposition and so on. So to conclude, and if we look back at our hypothesis, biodiversity do increase over the monitoring period. That was quite positive conclusion already. For the second hypothesis, that aquaforest biodiversity develops positively relative to its pre-construction state. It's also, we have on the bird point of view already a family and guild structure that trend towards a mangrove type, but for the macrobenthos it's not yet comparable. And for the third hypothesis, that aquaforest reaches a viable and ecological state, it's really too soon to tell. 13 months is not enough. We still need real tests. It's happening right now, so the mangrove is being tested, and we should wait maybe 5 to 7 years to really draw conclusions if this project was a success or not. So to give some recommendations, I just focus on the main ones. I would highly recommend to continue the monitoring over several years because, as I said before, it's a living laboratory. There's a lot to learn with this new artificial ecosystem. I will also improve the bandwidth graph protocol to characterize better the subtidal community, and I would like to investigate the trophic links between macrobenthos and the birds, because the birds are there for a reason. They might come there to feed, etc. So that would be my recommendations. Thank you for listening.
Last discussion for today. I understand we okay this time.
Yeah, sure. Thank you, Diego, for an interesting presentation. I would like to go back maybe to the initiation of this aquaforest project. Can you just go back? I think I probably missed something. How did it all started?
On the side?
Yeah. Is this sand deposition? What was the origin of this sand deposition?
So it's due to urban development. The city of Guayaquil, which is the second largest city of the country, is developing a lot. There's also shrimp farming that is growing a lot in this region. So for all these reasons, sediments are being removed from their original places. Another reason for that is that, for example, Jan de Nul are doing the maintenance of the access channel to the port. So there's a lot of sediments moving around, and Reno et al. Observed that the tidal flats were formed due to anthropogenic reasons. They were not there before this urban development.
Is it due to the urban development, or is it just natural sediment movement?
No.
In a estuarine system, the fact that you have tidal flats that create and disappear is pretty normal. No?
Yeah, but that, according to this study, it was really due to this.
Okay, so then we had this tidals flat, and then Jan de Nul arrived and decided to develop a mangrove on it.
Exactly.
Okay. What for?
It was a way to valorize their sediment waste, because they're doing the maintenance, they're dredging, And instead of just putting the sediment offshore or just somewhere else, they wanted to develop a new framework to maybe use this in the future for other projects. So that's why they decided to invest in this project and to create a.
So I understand that a company like Yadnul can say to avoid wasting this sediment, but scientifically speaking, that's like a bit shocking.
Yeah.
Sediment is never wasted. You know, the sediment moves and comes from somewhere, goes to somewhere, and it's never wasted. From a human-centered point of view, can consider that, but I think it's important as a scientist to be aware that it's all natural movements that arrive with or without the anthropological context.
But can I?
Yeah, absolutely.
Yeah, I was dubitative about this project at first, about the ethics of the company. Of course, it's a big engineering. They're there for the money and so on. But I thought that in the end it was really first it was turning, because they destroyed an ecosystem. There was a tidal flat that was sitting there. A mudflat ecologist would say, "You're destroying a mudflat." I don't agree, and actually it's just.
That was my next question.
It's just like turning a tidal flat into a more biodiverse ecosystem, and that's why I was talking about all these ecosystem services. Like a mangrove can enhance the fisheries on a local scale because it serves as juvenile nursery, and also just for science, I think maybe there was other option in this specific case, but maybe somewhere in the world that's the only option.
I agree with you that it's a very interesting natural laboratory. Should we always aim for higher biodiversity in ecological restoration?
No, I don't think so.
And what's biodiversity?
I think we should aim for the most adapted ecosystem. Yeah. If we restore an ecosystem, let's aim for the most adapted ones, and that's why also mangrove was chosen, because it's all over the place in the city.
If it's the most adapted, we don't have to restore it. No?
Yeah, but it was not there at the beginning. That's why. That's why. Yeah.
Okay, that's it for me. Thank you.
Yes, I hope that this new strategy is bringing hopes. So, on your developing mangroves, do you have an idea of the time it will take for the sediment to reach another organic carbon concentration that can sustain the benthos to be something comparable to a mangrove? Like, because the little things, the trees are small, and how many years it will take so that the tree will be large enough so that their detritus and leaves and whatever will create enough carbon so that really the sediment will change and will be considered as efficient to store the carbon?
That's a very good question. Honestly, it's difficult to answer in terms of like precise time, but there was a similar study done by my supervisor in Kenya, and they conclude that it's like 5 to 7 years or even more to really get this type of ecosystem services back. It takes a lot of time, and that's why I suggest to really continue the monitoring. But yeah, it's more, we're talking more about maybe 5 to 10 years or maybe even more.
10 years would be short.
Okay.
You know, it's like the whole cycles of falling leaves, and it means the trees have to really raise.
Yeah.
So maybe that was my next question. Maybe this is what explains that in fact the birds are giving you a response rather than the benthos. And I wanted to ask you, what is the use of the new ecosystem that the birds are what what are they using it for? Is it only for breeding? Is it for feeding? Because there's what is there that was not
It's
present before to feed them?
yeah, it's like for breeding, as you saw on the picture.
Yeah.
But it's also to feed. Like I haven't been on site. I didn't go to Ecuador, but my supervisor who went told me that there was an incredible amount of fiddler crabs, and this it was not characterized by the quadrants because they're super active on the surface. They're like the only ones that are inside my dataset, and that's a limitation of my research, is the ones that were dead, and they counted as part of the quadrats, but the thing is, on the inside of the site, like they sampled here on the basically on a small on a dike, it's more elevated, and if you look here, like that's where they sampled, but here it's super muddy and it's super it's not really accessible, so that's why they didn't go as far, but the entire surface there is full of fiddler crabs, and that's why it's one of my limitations. Yeah, I'm almost sure that they are there for a few.
Okay. That's the organic carbon coping.
Yeah. Sorry?
I said that's the organic carbon coping.
Yeah.
Okay, I don't know if I have another question, but yeah, I leave the floor to my colleagues. Thanks a lot.
Thank you. Olivier?
Thank you, Diego. I have some questions because there's some things either I missed or it wasn't said. In the data collection, you described basically the engineer site, and you only mentioned that, but then later you're comparing your results to some reference sites that you did not mention before. So what are these reference sites, and how were they sampled?
So the reference sites B1, B2, B3, B4, they're all around aqua forest.
Yeah.
They're all major mangrove, and they were selected based on different criteria, but one of the main ones was accessibility and if they were developed or not.
Okay.
And one of my limitations was that they were sampled with transects, so that's why for the bird analysis I use Jacquard dissimilarity to look at, because I only had qualitative data on the references. There was only presence absence. And for the macrobenthos samples in the reference mangroves, it was done using Van Veen graphs. And that was my main issue.
But the sampling design was the same? It was continuous through time at the test and the reference sites?
No, the sampling in the references was done in 2023, whereas the sampling on Aquaforest was done in 2025.
A single visit to the sites, or?
And that's one of my other recommendations that I didn't cite, but I was on myself. That's a big problem. They went just on a single survey. For me, it was not enough.
Well, that means when you're telling us that the richness of birds, for example, is higher at the aquaforest site, that's a whole year of counting compared to a single visit.
No, it's so like when I compared the references to aquaforest, I compared May 2023 to May 2025. So that's both a single day because it was only one monitoring survey per month.
Okay.
So that was the thing I wanted to highlight also in the presentation. To me, one single visit at the references is not enough to characterize entirely the reference mangrove, and also I think they should avoid have interannual variation during these sampling surveys. But to avoid for like migratory turnover, I chose to work with May.
The same season.
Time of the year, yeah.
Okay. I understand that you have like quantitative data for your benthos and presence absence data for your birds, but there is community data, right, in both cases?
Yeah, presence absence for the references. But for the aquaforest site, I have.
Yeah, but when you're dealing with beta diversity, you're putting everything together, so you're treating it as present absence even for the aquaforest site, right?
Yeah, for the birds, yeah.
Yeah. So, but what I don't understand is like, okay, you have quantitative data on one side and presence absence, so you cannot say use the same measures of beta diversity because you don't have the same information. But I don't. Can you have reasons to not have the same like methodology for both and not asking the same questions? Because you're using metric and non-metric. You're decomposing into nestedness and turnover, and you're not. What's the rationale behind doing these completely different steps in the analysis? Well, it's the same kind of data. There might be one, but I didn't get it.
I think the main issue was that the monitoring was done in the references and on the baseline, and the baseline study was done by local experts using their framework, and after the framework.
I'm not talking about the acquisition, I'm talking about the analysis and the questions you're asking. Like, to be more explicit, on the metals you use NMDS, and on the birds you use PCOA.
Yeah.
Why?
Because I thought like that NMDS was more adapted to abundance weighted data and PCOA for more. Because the PCOA is like I use taxonomic distances.
You use Jacquard.
Yeah.
Yeah, and the other kind is Ray Curtis, which is the same thing, but one uses quantitative data and the other one presence absence. So I just don't understand, and so I'm just wondering, is it because that's what you're used to?
No, I did my research and thought I understood that it was more adapted to each situation, because my analysis is really like separated into different groups.
I'm not sure that was the best choice, and if you're trying to, if the aim is to publish this, it's going to be, you're going to need to justify why the same type of data and the same kind of questions are addressed with two philosophies of analyzing this data. Because you can do like the same kind of also partitioning and turnover and nestedness or species difference or richness difference. You can do the same with Ray Curtis as we did with Jacquard. And would it not be interesting with the Bentos? So you can, you need to shut me up. Thank you very much, Diego.
Think we'll see beyond the.
Well, discuss over here.
Yeah.
Luana, do you have
I can wrap
maybe?
up. We'll try, but I want to acknowledge congratulations, Diego. Really enjoyed your presentation. You think you have excellent presentation skills. Also brings hope, because you mentioned now during the discussion with the jury the importance of. We think it's very good to have scientist collaboration in this. We have two, not more, because IMBC is a consortium of universities carry all the influence with you, Ben with as well, and the company collaborating together, but that you do it in a conscious way, and that you addressing this, it's extremely important. And as a good example for other collaborations of science and industry. Of question, did you have the chance to think of other bioindicators of relevance besides the birds and the macrofauna for the monitoring?
We talked about characterizing the subtidal community, more looking at the phytoplankton. But apart from that, I don't really know. That was our discussions after I submitted the manuscript. But they were talking about characterizing, because like the subtidal, the Van Wingard methodology is not working. They're going at different tide levels, so that's why the data is quite poor. And then they were talking about have a look at the phytoplankton, but I will not say.
Of course, here we are two bioauthentic specialists.
No one
And
wants
of
to talk
course,
about
they are
Right.
as a recommendation.
It's very hard,
Despite the prejudice of the jury. It would be good, strong advice to pass along for the project. Being important, it's the chances of success for monitoring under these conditions, especially when the sediments are higher. So it's as a recommendation. You hear it's not uncommon that people overlook the neobentic compartment, but there is an advantage to add to it, and we will overcome the constraints and just if the two we will serve for the macrofauna. There are some also a device to look upon the fauna associated with the plant species.
That's something like the projects is going to continue, of course. Or it's also a first year that's going to continue thesis, and that they were talking about that. The only thing that it was I didn't have the information about plant development. I do know where which species are planted, but I don't have a clue of the development of this plant. So that's definitely a barrier that we need to look at.
So we understand it's a 6-month period for the master thesis. It's again for to enrich the discussion and hear about your insights on it. I hope the aquifers will survive this. Appreciate you being aware of that phenomena, especially this year that is happening. I don't have further questions.
Thank you, Luana, also for advocating for the marijuana community. Indeed, it's very rich marijuana community in the mangrove sentiments inside. It works in mangroves for marijuana community, but there is a special practice. If I come, I will serve it. Congratulations.